Welcome everyone to our webinar. We'll begin shortly. We'll let some folks come into the virtual room. Welcome folks. As you're coming into the virtual room, we'll just give a few more seconds to let a few more people come in before we get started. Okay, all right. Good afternoon, everyone, and welcome back to day 2 of the FDA Duke-Margolis workshop on advancing drug development for the prevention of spontaneous preterm birth. I'm Nancy Allen Lapointe, a faculty fellow at the Duke-Margolis Center for Health Policy, and I have the pleasure of opening and closing the second day of this workshop. Just a reminder, the workshop is being recorded, and that recording will be posted to the event webpage. To set the stage for today's session, I want to very quickly recap the discussion yesterday. Yesterday, our speakers highlighted the urgency and importance of addressing preterm birth as a serious public health problem that affects millions of lives. In the patient-focused session, we heard the compelling stories of parents and frontline staff from their experiences in the NICU. In the other sessions, the experts discussed the increasing and unequal rates of preterm birth and the need for more research on its biological mechanisms and potential treatments, and explored the ethical and regulatory issues of developing therapeutics for preventing preterm birth, such as the lack of products for obstetric conditions, the exclusion of pregnant persons from research, and the challenges of measuring fetal effects and clinical outcomes. The speakers also suggested some possible solutions, such as more funding, infrastructure, consensus, and innovation to overcome the barriers and advance the field of preterm birth prevention. So with that, let's turn to our agenda for this afternoon. This afternoon, we're going to have 2 discussion-based sessions that will hone in on important considerations for the development of the prevention of spontaneous preterm birth. The first session today will focus on efficacy and safety assessments in therapy development programs. And the second session today will focus on clinical trial design considerations, including dose finding. Then we'll end the afternoon with a discussion among some of our workshop moderators and panelists from day 1 and 2 to provide their key takeaways of the workshop and key next steps. As Mark and Rob said in their opening remarks yesterday, the purpose of this workshop is not to focus on McKenna, as that is outside the scope of the workshop, but rather to focus on looking forward and advancing the development of new, safe, and effective therapies for preventing preterm birth. We are really honored and pleased to have a distinguished group of expert stakeholders today from the FDA, academia, and industry to drive this conversation around advancing drug development for the prevention of spontaneous preterm birth. As a reminder, you can find more information about our speakers and panelists in the bio sheets that are available on the event website. For all of our attendees, thank you for joining. We are happy to have you here and encourage your active participation through submission of comments in the Zoom Q&A feature. We will try to incorporate as many of those as possible in the discussions. Now, without further ado, I'd like to move to our first session for this afternoon and introduce our presenter, Lynn Yao from the US FDA, who will be moderating this session. Lynn is the director for the Division of Pediatric and Maternal Health in the Office of New Drugs within CDER. So over to you, Lynn. Thank you.
Thank you so much, Nancy. It's really an honor to be here, and I'm just very pleased to be able to hear the discussions in this session. I will start out by saying, if we could have the next slide. This session is really going to be focused on, perspectives from all of our stakeholders, regulators, academicians, researchers, and industry, on how we should think about efficacy and safety in clinical trials to evaluate spontaneous preterm birth, and to address the challenges involved in running these clinical trials. I think importantly, we hope to get out of this session how to identify clinically relevant neonatal outcomes And based on yesterday's discussions, I think we really need to ensure also that we're enrolling the correct populations. So I'd like to start out by introducing our panelists, who we'll give them a few minutes each to provide opening remarks. We'll go in order, and then after each of their remarks, we'll go ahead and start the discussion. And first up, I really happy to introduce Dr. Mark Hudak. Mark, take it away.
All right. All right, so my video is not starting for some reason. Start my video. Okay, does that work? Great. So Lynn, great to see old friends. Thank you. Good afternoon, everybody. I am pleased to lead off this session 4, and I hope not to overlap at least too much with my fellow panelists. I was asked as a clinician and investigator to offer some comments on what may constitute meaningful and feasible outcome measures in neonates in the context of therapies to delay preterm birth, and with the added perspective of what we have learned from the McKenna trials. So in the McKenna trial, the FDA used a reduction in preterm birth as a surrogate for improved neonatal outcome when it granted accelerated approval based on the first study. The rationale was that it was reasonable to expect that prolongation of gestation should result in improved neonatal survival and reduced neonatal morbidity. Now, of course, the magnitude of these effects, if any, would depend critically on the differences in distributions of gestational ages between the placebo and the treatment groups, with particular attention to the outcomes of those most vulnerable, specifically infants born before 29 weeks gestational age. The FDA did recognize that in some cases, prolongation of pregnancy might have a deleterious rather than a salutary effect on the fetus and newborn, and required comprehensive assessment of neonatal outcomes in trial 03. So we know that under extraordinary circumstances, prolongation of gestational age may result in a worse neonatal outcome. This is something my OB colleagues struggle with, for instance, when deciding the optimal time for delivery in the face of uteroplacental insufficiency that is compromising fetal growth or some other indicator of fetal well-being. Now, I should state that gestational age by itself is not necessarily the best predictor of survival or morbidity. For instance, you can go to the NIH website and use their neonatal calculator. That will estimate that the average survival for a 24-week, 800-gram singleton female with exposure to antenatal steroids is 84%. That would compare to a 24-week multiple birth male infant not exposed to steroids survival average 18%. Nonetheless, on a population level, every additional week of gestation does confer advantages, including expectations for higher survival, a lower likelihood of short-term morbidities such as BPD, severe IVH, ROP, and NEC, and a lower risk of longer-term morbidities such as cerebral palsy and cognitive delay. Importantly, each morbidity has a different profile of risk versus gestational age. There are different windows of risk for each of those morbidities and different highs and lows of incidence by gestational age. In trials of neonatal therapeutics, randomized control trials with or without stratification do ensure balance of infants by major risk factors, so that the demographics of treatment groups are usually quite comparable, especially with large trials. This equal footing, if you will, of treatment groups allows us to have confidence in evaluating primary efficacy outcome, secondary efficacy outcomes, and safety outcomes. Nonetheless, a divergence of primary efficacy and secondary or safety outcomes has occasionally presented challenges in deciding how to act on the outcomes of the trial. Composite outcomes in randomized controlled trials in neonates are tricky, and these do lose utility when they weight each morbidity equally. This was the case in McKenna trial 03 because the composite adverse neonatal outcome was a yes or no. It was defined as death and or the presence of any one of 5 morbidities. Efforts to develop effective treatments to prolong gestation in women at risk of preterm birth do pose special challenges. First, because the mother-fetus dyad is the subject of the trial, The trial must confirm no harm to mother, no harm to fetus, and not produce a worse outcome for the neonate, the child, the adolescent, the young adult, and the adult. So long-term follow-up in these trials is especially critical. Second, if a trial does prolong pregnancy, what are the reasonable expectations for newborn outcomes? Should survival be the same or should it be higher? And if so, by how much? And how would you determine that? Should neonatal morbidities be the same or lower? Again, by how much? And how would actually one quantify that? And how about later outcome in childhood? We know that DES had generational effects. Also, some of the animal data on Makina suggested adverse effects on fetal rat brain development. And we do know that some interventions in neonatology can be harmful. For instance, there is moderate to high quality evidence that early use of systemic dexamethasone in preterm babies at risk for BPD causes harm. Not because it changes BPD in a bad way, it did reduce BPD, but we belatedly recognize that it also increased the risk of cerebral palsy in infants at 18 months corrected age. So as a modest suggestion, I would say that we should do a different type of analysis for neonatal outcomes in a trial that might shift the distribution of weeks of completed gestation between the treatment groups. It seems reasonable to compare the distribution curves of survival and each morbidity by gestational age for the treatment groups. Similarly, composite outcomes can be weighted and analyzed similarly. So I think there are some opportunities to better sort of define what the neonatal outcomes are and to compare them in a more meaningful way. And I'll end my remarks there.
Thank you so much, Mark. That was a lot packed in to 3 minutes. So we'll try and unpack it a little bit more during the discussion. Next, we would love to hear from Tamara. And again, I just want to say we have a stellar group of panelists today, and Tamara's one of those. So, and you can read those bios, as Nancy said, in the packet that is on the website. Thank you, Tamara.
Yes. Hi, everyone. I'm Tamara Lewis. I'm an academic neonatologist and pediatric clinical pharmacologist at the University of Toronto. For the past 10 years, I've worked as a neonatologist in level 4 NICUs at big academic children's hospitals. So I'm very honored to be part of this 2-day session. And I wanted to start by saying that my comments in the next few minutes and during this panel are not meant in any way to criticize the really important prior efforts in clinical trial design and execution in this space of prevention of preterm birth. So I hope that our comments are not seen in a critical way, but in a really constructive way. You know, I think from hearing the discussion yesterday and building upon Dr. Hudak's comments, it's pretty clear that using a pure gestational age cutoff or composite neonatal outcomes that are really binary in nature, did you have this morbidity, yes, no, are not sufficient to capture the nuance of outcomes in preterm birth. And so I think it's important for us to use this time to really think outside the box and think quite expansively about the ways that we can measure the morbidities of preterm birth. What also struck me from yesterday is how critically different our current clinical trial outcomes are from what actually matters to families of preterm infants. The testimony of families and the social worker yesterday was very powerful and I think was a call to action for us as a research community to reconsider what is important to quantify in clinical trials. So at the end of the day, everybody on this call who's working in this space, our real goal is to have an intact mother-infant dyad. That is what is truly important to clinicians, to families, and regulators and drug developers. And this concept of an intact mother-infant dyad is something that seems quite nebulous, but I think has to be the driving force behind the way we design our clinical trials. One thing that I think is important for us to consider as a research community is that we have to consider different gestational age brackets as different levels of morbidities. And I think that we have to define our neonatal outcomes within different gestational age brackets instead of trying to apply the same neonatal outcomes across from a 22-weeker to a 36-weeker. And one approach may be that within each gestational age bracket, we have time to event clinical outcomes. And this would align very well with the things that the family shared yesterday were very important to them. So time to important NICU milestones. So for the very preterm population, let's say 22 to 26 weeks, one important outcome may be time to final extubation from the ventilator, or time to the ability to try oral feeding for the first time. And of course, these neonatal outcomes that are more nuanced, not just binary, can be influenced by local clinical management. But I think that we have been able to do big RCTs with nuanced outcomes in other disease processes that are influenced by local clinical care, but we still count those outcomes as valid. Then we could compare, similar to what Dr. Hudak said, the distribution of these more nuanced neonatal outcomes between the placebo and the treatment arms for these preterm birth trials. Another important endpoint to both clinicians and families is inpatient length of stay. And of course, we can nuance this primary clinical trial outcome to account for gestational age at birth and other important confounders. But I think length of stay is a really important overall marker of morbidity for these NICU infants. And then I agree that we have to somehow capture the extent of medical morbidity at NICU discharge. Each of the parents who testified yesterday talked about how this saga doesn't end for mothers of preterm infants at the time of NICU discharge, and that it was the things that happened when the babies were 1 and 5 and 7 years old that were equally important. And so I know it adds cost and length to clinical trials, but really we have to come up with ways to quantify medical morbidity at the time of NICU discharge. I think really focusing on 2 very important organs, brain injury and BPD. And then really the extent of ongoing medical morbidity at key early life milestones, for example, 2 years of age. The other thing I'd briefly like to comment on is that as researchers in general, for the way we measure confounding variables in clinical trial outcomes, we've really taken a reductionist approach. I think that's out of necessity. We're gravitate towards things that are easy to measure and easy to quantify in a standard way. But that really leaves us trying to investigate a 4D problem like preterm birth with 2D tools. And so I would encourage us to move away from that reductionist approach, especially because when we do that, we try to interpret drug effect in a vacuum of all of the important things that influence preterm birth outside of this drug that we're testing. So we need a lot of more nuance and rigor in the way we're measuring confounding factors like the experience of lived racism, social stressors to mothers, all things that can really affect our outcome in addition to our RCT drug, but that right now we're not collecting in a nuanced way. So I'll stop there. Thank you.
Thank you so much, Tamara. Really, I think, important insights. I think that we're going to have a pretty robust conversation because I'm already running out of space on my note taking just from Mark and Tamara's comments. But next, I want to turn to the other side of the delivery, Anjali Kaimal, who will also give remarks as a maternal fetal medicine specialist. Thank you, Anjali, for joining.
Let's see, can you guys see my video? I'm not seeing it for some reason.
Not yet.
Having...
Yes.
Okay. All right, there. Excellent. Very good. Sorry for those technical difficulties. So thank you so much for the opportunity to participate in this, as we said already, very robust discussion. I think the speakers yesterday laid out many of the issues that are important in thinking about meaningful outcomes for future studies. And my neonatology colleagues, Dr. Hudak and Dr. Lewis, comments today further highlight some of the logistical issues that are truly challenging to consider as we think about planning for future research. I know we're going to have some more opportunities to discuss some of those specifics, so I wanted to just share a few broad ideas to highlight from my perspective, both as a maternal fetal medicine specialist practicing clinically and as a researcher who focuses on patient-provider counseling and the implementation of evidence-based care. So turning to some of the speakers yesterday, Dr. Elevitz's discussion of phenotyping, I think, is so important. We really know that preterm birth is not a monolithic disease in any way. And thinking about approaches to treatment without understanding the different pathophysiologic processes that lead to an early delivery just sets us up for negative results. So this is really hard to do, but just essential before we think about that next step. Turning to the other end of the research spectrum, where most of my work focuses, I would urge really early consideration of the ways in which an intervention would ultimately be accessed at a population level. We should think about the barriers to equitable care and how to overcome them upfront. And as Dr. Lewis just highlighted, we know that real world conditions also impact efficacy. So if we haven't taken those things into account, we may end up in a space where we have a treatment that is not able to deliver what we thought it could based on the way that we designed our original trial. I think yesterday, the patient perspective, both in understanding the short and long-term outcomes and also understanding what is clinically meaningful, brought really essential context to this conversation. It also ties back to that idea of the discussion of phenotyping, different pathophysiologic processes, and what are the outcomes that are important. I would love to say we're going to cure preterm birth, but I think we all know that that's unlikely. And so when we think about what the outcomes are that we can consider, we know that clinically, a week prolongation of pregnancy at 24 weeks has very different implications than a week prolongation at 34 weeks. But the value that patients assign to those and the outcomes that come from them may also vary. So even at 34 weeks, a prolongation of a week may lead to a meaningful difference in the patient experience in terms of those other outcomes we talked about, length of stay, approach to feeding, rooming in. And we need to think about how to incorporate those considerations when we want to target what are the investigations we focus on moving forward. I think feasibility of measurement and length of follow-up when we think about our outcomes is an important thing to consider, both in terms of what we would like to hope for and that what we can really expect realistically. As we heard from patients yesterday, preterm birth has lifelong implications, but engagement in research is not always easy. And we need to think about how we are incorporating the idea of what's important to patients, but also making sure that when we design a trial, we're able to offer the opportunity to be able to participate in that long-term fashion so we can get all of that information we need. I want to thank Dr. Lyerly for her reference yesterday to some of my comments at the McKenna hearing. I think hopefully we're moving into a time when we can do more research that's really focused on therapeutics specifically designed for pregnant people. But I also think it's really important to consider some of the issues she brought up in terms of reframing our discussion about the risks and benefits of both intervention and non-intervention in all of these different clinical contexts. We all know that when faced with a patient asking for help, not being able to offer any treatment feels really untenable. That is not what any of us want to have to do. And it's a very uncomfortable place to be, I agree. But until we have evidence for the treatments we're offering, we have to be honest in discussing the limitations of our knowledge. And that includes as we're designing trials and thinking about how we would offer these interventions in the setting of research going forward. And I'll stop there.
Well, wonderful, Anjali. Thank you so much for those comments. Again, I think, and thank you for tying in many of your comments with the presentations and discussion yesterday. Next, I'll turn over to one of my FDA colleagues, a dear friend and colleague of mine, Christy Chang, who is the director of the Division of Urology, Obstetrics, and Gynecology. Thanks, Christy.
Thank you, Lynn. So I really want to thank everybody for attending and for all our speakers yesterday and today to give such thoughtful remarks and very insightful comments. Having heard from Mark and Anjali and Tamara, I think I would rather keep my comments short because we want to dedicate so much time, a lot more time to listen to what these experts have to say. Currently, as everyone may recall from the McKenna trial, we had two co-primary endpoints to assess efficacy. The first one being the gestational age of delivery. The second one was the composite neonatal mortality and morbidity endpoint. But having heard all the discussion yesterday and from the comments in the session, we're recognizing that those may not be really clinically meaningful and may be suboptimal and not adequate to capture really what's important to our families and the children that we're trying to care for. So I think I'll end here and just give my time back to discussion, if that's okay.
Christy, always thinking of others. Thank you so much for your comments. And I will go directly to my next dear FDA colleague, Ann Massaro, who is a neonatologist and part of the Office of Pediatric Therapeutics. So Ann, please.
Thank you, Lynn, and thank you for giving me the opportunity to make a few comments. I'll just say, we have a bunch of neonatologists on this panel. I'm a neonatologist and spent, nearly 20 years in academic neonatology before coming to the FDA. And there's the old adage, you get 5 neonatologists in a room, you'll get 5 different answers to the question. But I think we have actually several common themes, I think, across the comments that I'll make and that Dr. Hudak and Dr. Lewis. And I think we have another esteemed neonatologist to follow my comments. But I think we have some common themes here. I'd just like to speak from the regulatory perspective and expand on some of the considerations that Dr. Chang discussed yesterday when it comes to endpoint selection for a trial of an investigational product intended to prevent or prevent preterm birth. As a neonatologist, one of the tenets of our teaching is that there's this inverse relationship between mortality, morbidity, and preterm infants and gestational age. And as Dr. Hudak mentioned, the different numbers on the X and Y axis may be different based on the different morbidities, but we all see that same bar graph or that same curve that, the lower the gestational age, the higher the rates of these individual morbidities that were mentioned, NEC, BPD, ROP. And so it seems logical to say that if we want to prevent preterm birth, and looking at prolonging gestation as measured by gestational age makes some sense. But from the regulatory perspective, this can be problematic for several reasons, just relying on gestational age. And some of these points were highlighted by Christy during her talk yesterday, that statutory basis we have for drug approval really rests upon having substantial evidence of effectiveness in a clinically meaningful endpoint. And that's if how a person or a neonate feels, functions, and survives. So gestational age, we know, doesn't meet that standard. So if we look at gestational age as that surrogate endpoint, again, we want to consider that HHS and FDA framework for surrogate endpoints or biomarkers, where we need to accept that correlation between gestational age and neonatal outcome to be so strong that gestational age can be a validated surrogate endpoint in its own right. And I think for the reasons that we've been discussing, and Dr. Hudak mentioned as well, we know that those there's so many different factors that go into the individual morbidities of preterm birth that, relying on gestational age alone becomes really problematic. So that other critical point, I think, raised by Dr. Chang during her talk yesterday and mentioned by some of the panelists already, is that all of that data that we have looking at that correlation between gestational age and the morbidities of preterm birth really are in the setting of spontaneous preterm birth, and whether drug-induced prolongation of gestation is associated with those same benefits is really unknown. So, and then the other issue with gestational age that, again, has been touched upon is, where's that appropriate threshold? Is it 37 weeks? Is it 34 weeks? Is it 28 weeks? We know that the most extreme preterm birth is luckily a rare event, but obviously with the most severe outcomes and something that we greatly want to avoid. But how do we weigh that or assess that in relation to prevention of preterm birth at later gestations? So this all goes back to really needing that clinical measure or clinical outcome measurement of neonatal outcomes and some overall measure of prematurity related morbidity, because looking at the individual morbidities by themselves is really challenging or difficult in the context of a trial. So, as Christy mentioned, co-primary endpoint approach with looking at an overall index of neonatal morbidity has been what we've, historically or relied on or thought about, because it seems relatively straightforward to know the different comorbidities or prematurity and put them together in some kind of index. And the selection of that index is a whole area that I hope we'll be able to discuss some during the discussion period. And different ones have been proposed, by the MFMU network, morbidity, mortality index, some other neonatal outcome indicators have been discussed. But again, all of these largely are different types of composites that weight these different individual morbidity, prematurity associated morbidities equally, which, as we heard from the families and panelists yesterday, is probably not the best way to look at these types of outcomes. I'll note, for folks who are interested, we did have another Duke-Margolis meeting back in the spring on neonatal endpoints for neonatal clinical trials. And a lot of that discussion may be applicable here to this discussion as well, where we talked about different types of endpoints and discussed, the idea of using a global rank scale that's been used successfully in the adult cardiology world and is, being developed for to develop a global rank scale for neonatal outcomes as well. But obviously that's still a work in progress, but something that might help in this area in the future. And again, when we're talking about weighing and putting together all of these different morbidities, it's really imperative to have that multi-stakeholder input, and particularly families, family advocacy groups and organizations, because they're really the ones who know, what weight to attribute to these different outcomes that we're measuring. So I look forward to the discussion from the panel to come up with, to talk about some of these ideas, and obviously, hopefully come up with some new ideas that are clinically reasonable, but also align with, the regulatory requirements that we have to work within. So I'll stop there and turn it over to our last speaker.
Thank you so much, Anne, for your comments. I think you highlighted sort of the tension between, what we think we can do or what we should do, and how does that fit into, the framework, right, from a regulatory perspective that we must operate under when we're thinking about development of new therapies. And so that leads me to our last panelist for this session, a former FDA colleague of mine, a current friend and colleague of mine, Robert Skip Nelson, who is trained in critical care, neonatology, and ethics. And I'm really anxious to hear some of your thoughts today as well, Skip, so please take it away.
Yeah, thanks, Lynn. And whatever I was planning to say when we had our pre-meeting, I'm no longer saying. I'm going to be responding to thoughts that came up yesterday, and I think it dovetails nicely with the comments that people have made so far. But let me first start off with a comment about why is there limited interest in industry on this topic? And I don't think the answer there is very difficult to ascertain. There's no clear understanding of the biological drivers of spontaneous preterm birth, and all of the etiologies of preterm birth are being lumped together. It's not monolithic. Bottom line, it doesn't sound like right now there's any actionable science to where one can develop a drug that would, or some other intervention that we heard a lot about the microbiome, and that may be promising. But until we have actionable science, you're not going to see a lot of industry interest. That's the bottom line. And so I think the focus of the field should be trying to develop actionable science to be able to decide where those drug targets are and how you divide that population. That's a whole separate topic. Let me turn my comments then to the neonatal endpoints. And this was stimulated large part by Christie's presentation yesterday about the gestational age not being proven, and that the drug-induced prolongation of pregnancy may or may not be a good thing, depending upon the quality of the intrauterine environment. Is it a good thing to stay in or a good thing to come out? As Mark mentioned, that's a dilemma that obstetricians and maternal fetal specialists wrestle with at all times. And suggesting that you do need a reduction in adverse neonatal outcomes in preterm birth to be able to get that clinical endpoint. But what would that be? I mean, there was a comment yesterday that 32 weeks spontaneous birth is not the same as a 32-week delayed birth through a drug-induced prolongation of pregnancy. But I would argue that that's really the wrong comparison. We're not comparing, let's say, a 27-weeker who would have been born at 24 weeks but had delayed to someone who is born at 27 weeks without delay. What we're really looking at is a group comparison of, let's say, all comers at 24 weeks, with one group maybe being delayed a week through standard of care and other interventions that obstetricians and MFMs are using now. Two, if a drug prolongs gestation to perhaps 3 weeks to go to 27 weeks. So it's really a group comparison of two groups, one with a little bit of prolongation based on whatever bed rest and other things that are done now, versus hopefully a longer prolongation. I might remind people, I believe Makina failed on the gestational age endpoint in its confirmatory trial, so the neonatal outcome in some sense was pretty straightforward. So if you think about this group comparison, you're really looking at group means. I think Mark mentioned this. Let's say between a group that has a mean gestational age of 25, having been delayed a week, that's the placebo group, let's say, or standard of care group, versus those that might be delayed to 27 weeks, maybe 28 weeks. And it occurred to me, as if you assume, and again, this is a big assumption, if you assume that there's no negative impact of the delay, meaning that the intrauterine environment at this point is taken out as a factor, and that's an assumption, it occurred to me that one could begin to model the very questions that we're asking around what that neonatal endpoint would look like by taking neonatal data and looking at 25 weeks versus, let's say, 27 weeks or 28 weeks, and asking, again, if the intrauterine environment is neutral, big assumption, what would these endpoints look like? BPD, ROP, IVH, cerebral palsy, what would it look like? I know, for example, that the Critical Path Institute with the International Neonatal Consortium is doing a real-world data analytics platform. They say they have 300,000 participants in there. I would also put out an appeal, if this project made sense, for other neonatal groups to put in data. Large commercial groups, such as pediatrics, could put in their data. Could one begin to model what that neonatal output, neonatal composite, whether it's composite or separate or however we do that? I agree with the weighting. Not all morbidities are created equal. Could we model what that would look like to where one could have some realistic assumption of what the sample size might be? Because the fear is, given the success of neonatal programs to date, is that that a neonatal endpoint, whether separate or composite, however we structure that, and I would agree, not just a yes/no binary kind of thing, could end up with a fairly large sample size. What does that look like? How much of a gestational age impact would you need to have in order to have a positive outcome on that neonatal endpoint? I think that could be modeled with existing clinical data. It's just a matter of pulling that together. And then if you had a negative intrauterine environment, that effect size would decrease, obviously, and a drug could end up, even if it extended the gestational age, if that intrauterine environment was harmful, you would have a decrease in that effect. But at least that would give you sort of the boundaries of what that trial might look like. So I think it's a challenge to do this, but I think we could potentially bring data to bear on trying to decide what that composite neonatal endpoint ought to look like. And that in and of itself, in addition to some actionable science, if people knew these trials were actually feasible that might begin to generate interest. So I'll stop there.
Thank you, Skip. And I'd invite all of our panelists to turn on their cameras now so that the group can see all of us. Wonderful, wonderful. A lot to chew on, and I'm not sure if my brain this afternoon is up to the task, but I'll try. So one of the things that I feel like I'm hearing again and again is that, and I think Anjali, you said it again, that was sort of related to what we heard yesterday from Michal, which was that, we need to understand the phenotype. We need to understand the different types of, patients who develop spontaneous preterm birth before we can hope to run a trial, because if we don't have the right population, it doesn't matter what endpoint we have, right? So knowing that, is there a population that you think we know already has particular characteristics that could be studied, or are we not there yet? Is the science just not there yet? I'll open it up. I'll ask Anjali first, but then I'd like to hear others on this topic.
I see Dr. Elovitz is also on today, so I'm sure she would have comments and thoughts about this, and she has devoted her life to looking at these questions. So I think it's a challenge, and I think it is a little bit also goes back to some of the discussion about that Dr. Lyerly had about the sort of the way in which we frame obstetric research, and how do we want to make sure that it is either similar or different to the way that we frame research outside of obstetrics. So I think we have some information. I think we have imperfect information, partially because we haven't focused as much on it, because we have been wanting to get to that next step of making sure that there's a treatment available. But I do think also that in other areas of medicine, there is kind of a combined approach where you're trying to do all of these things at once to a certain extent. And so my comments really are more about saying the phenotypical work has been going on, continues to go on, both in the lab and clinically. And how do we make sure that we're continuing to support that with a specific, sort of view of saying what we're trying to do here is really figure out what are the different groups that might have a target for therapeutics. They do that in oncology all the time. They do that in internal medicine all the time. And I think we should be able to do those things as well. I just think that if we only focus on therapeutics without thinking about what is the population of people that we think is able to benefit from this, then we end up enrolling this broad group of people that some of them might benefit, but all of them don't. And therefore you end up with a negative trial.
That's right. That's right. Thank you. And I see Tamara, please.
Hi, I wanted to ask a follow-up question to my MFM colleague. So I was thinking last night, are there ways that we could do trial enrichment, where we enrich the clinical trial population for women that are at extremely high risk for these preterm birth outcomes? And what I was thinking is there a role for focusing on women who have already had spontaneous preterm birth at a very young gestational age? The number we can debate, less than 28 weeks, less than 29 weeks. Every NICU across the country has these moms who've had these micropreemies. And, part of the issue is that if we were to enrich for this patient population, we would have to decentralize the trials, meaning we would have to allow women to enroll outside of, 19 academic centers. But that is a group of women that is extremely high risk, I think, and would enrich clinical trial population and success. So I'm interested to hear your thoughts on that.
No, I think that's a great question. I think also one thing we haven't focused on too much is, are we talking about recurrent preterm birth or spontaneous preterm birth in a first pregnancy, which obviously are two different things. I think trying to identify the population for a recurrent preterm birth is somewhat logistically easier. I think in going back to that idea about phenotyping, I think where we need to start is for those people who are presenting in spontaneous preterm labor or with PPROM, what's the information we can collect both clinically and biologically in terms of sampling and biobanking to make sure that we have the information we need in that first pregnancy when they're delivering. And then I totally agree with you. I think one of the things that's really hard about doing sort of implementation science science research is the idea that where actually most care is being provided for obstetrics is in the community. And so the places where you most need to understand how you're going to be able to enroll and then ultimately implement are not the places that generally are the centers that are submitting the grants. And so how do we sort of overcome the logistic challenges of doing that? And then obviously for something like very early preterm birth, just based on the numbers, in order to be able to enroll enough people who have those characteristics, you have to be able to include something beyond just the major academic centers, which I think overall the centers have done a good job of trying to enroll more community sites. We just need to continue to do that and do more of it. I think the last thing that comes up though with those patients who have had a very early preterm birth is kind of my last comment about how challenging it is to really admit how little we know when you're trying to enroll patients. So I think in the discussions that we've had about the withdrawal of, various different interventions that we've tried that ultimately did not turn out to work as well as we had hoped, a lot of what the discussion comes from people who are practicing clinically is how am I supposed to stand in front of my patient and tell them that I don't have anything to offer them? And then some of the discussion about withdrawal of Makena was, is everybody going to end up with a cerclage, which is an intervention that has more risks because we won't be willing to stand in that space where we acknowledge the limitations that we have. So I think that is part of, what we need to understand from our patients to say, we want to be honest with you. We want to be able to tell you this is where our knowledge stands and this is the way that we could get more information. But it does require us to sort of sit in this uncertainty together, which is not easy. But I think that's part of what the barriers are to enrolling that population who really have had those very significant outcomes in the prior pregnancy.
Yeah, thank you. Thank you so much, Anjali. I see Christina and then Skip. And then Mark.
Okay, hi. So I think as to how to enrich the population in the trial, I think that's a really a good idea and a good comment. And I just want to add that, as much as FDA has maybe critical of the MEAS trial, I do want to comment or commend the NICHD, the MFMU network for really committing resources to go out and do these trials. And one thing about enrichment, I just want to mention that, we may be willing to accept more uncertainties when, with a 28-weeker or 30-weeker, when it comes to whether the gestational age of delivery really translate into the clinical outcome, and whether benefit has been shown versus a 36-weeker. So, how can we weigh the different gestational ages? And that's a topic that I really like to hear from. Thanks.
Thank you, Christy. So Skip and then Mark.
Yeah, I guess one comment stimulated by Tamara's suggestion, and then maybe a response to Christy. Enrichment is a good idea if you have some idea of the target that you're going after. Behind that idea is an assumption that you have some knowledge of the pathophysiology and you know that what your intervention is going to do is have some impact, in fact, on that pathophysiology. And from what I learned yesterday, I don't see that knowledge. And so at this point, to start talking about enrichment is putting the cart before the horse. I really think you have to have some idea of the target as the target present. If you know the target's there, you don't need very big trials in conditions that affect pregnant persons. You can do trials if you have some idea of a druggable target. I also agree that, yes, I would anticipate that there ought to be a higher effect size potentially in the 24 to 29 week period than you might see in the 34 to 36 week gestation, only because of the differences in morbidity and mortality that exist within those. But again, that's something, intuitively that makes sense, that it would be easier to see an effect on neonatal outcomes in that lower gestational age. But again, that's where I think we need to do some of the modeling to see whether that's in fact true, and just take neonatal clinical data and run an in silico trial to see what those differences might be in some of those composite outcomes.
Yeah, Skip, hold that thought because I want to come back to it, but please, Mark.
Yeah, so a couple reactions to what people have been saying. So I think it's worthwhile going back and looking, realizing that in the big Makina trial, the percent of women who gave birth before 32 weeks was only 5%. So when you talk about enrichment, I think that demonstrates, because I do think they try to enrich that population to try to capture the highest risk women for delivering preterm, and I think it's just hard to do. Angela, I'm not an OB, but my impression over the years is there are certainly different phenotypes that operate for the very preterm births. So you get a population of mothers who come in and they've had, cervical shortening and relatively painless cervical dilatation, and 24 weeks out pops the baby. Compared to a mother who comes in with, grinding labor at 25 weeks. I mean, I think those are probably different etiologies and mechanisms of preterm delivery. And it's hard to predict who's going to come in and deliver preterm. I mean, you got a high risk group, but, they're not guaranteed to deliver preterm in the future. And most of the mothers who come in and deliver preterm have no history of preterm birth, at least in my experience. So, I think it's really hard. I think, just to your point, Skip, a composite outcome, I think, can be constructed. There's a lot of data out there. Vaughn just published a paper this month in Pediatrics that's really powerful in terms of it shows all of the morbidities and survival by gestational age, by over time. So it shows you what's happened at each gestational age over time. It shows you the difference in outcomes at any time with respect to those outcomes. I think those data are there. I mean, they're large data sets. We could look at those sort of things. I think that's really important, and those things can be weighted, I believe. And then to Maura's comment, I think the length of stay issue is really important too. It's a functional issue. It's something of impact to families. I would probably do it a little differently. I'd probably look at the distributions of postmenstrual age and discharge as a function of gestational age, because we know that, if you look by gestational age, you have your, long, high postmenstrual age and discharge for babies at 23, 24 weeks. Those come down. They reach a minimum of about 36, 37 weeks, and they start going up again because you're delivering babies now who are 40, 41 weeks. So those can be modeled, and I think that is an important short-term outcome for parents for sure.
Thanks, Mark. I don't see any hands up, so I'm going to ask the follow-up question that I wanted to ask. So, right, what I'm hearing from the group right now is that, look, delivering at 24 weeks even, although we know that that's a population that we, will expect to see time to event morbidities, much more likely than if we take all comers 24 to 36, 34 weeks. But even in that group of 24, Mark has described, and I think we heard yesterday, that even, those infants who deliver at 24 weeks don't constitute necessarily the same type of reason for delivering early, and therefore maybe we're missing the boat here in terms of lumping them all together. So what at a minimum, I would ask the group, do you think, what's the research that has to be completed before we feel comfortable in running a trial at all, with whatever endpoints? And I think Skip's point is well made about endpoints. Look, if we want to figure out what the best endpoints are, we got to take all the data that we've collected. We got to model it. We got to take into, I think, the important points that Tamara made about, taking in what we heard yesterday from our patients and families. But it doesn't sound like we can even start a trial is what I'm hearing, even if we had a great endpoint, which I don't think we're there yet either. So I want to hear, what else do we need to do to identify the population that we think we could go into, the one that's maybe most likely going to have, an outcome or something that we could do in a feasible amount of time. So I see Skip's hand, and then I'd like to hear from others.
Yeah, Lynn, you made a comment. I just want to make sure it was clear. I don't think there's any data. I mean, Mark could maybe correct me on this, or Tamara, that would suggest differences in neonatal outcome depending upon, let's say, the differences in the causes of your preterm birth, let's say at 24 weeks. one 24-weeker compared to another 24-week relative to what may have been the underlying etiology of the cause of that preterm birth at the same gestational age. Maybe there might be some differences, but I haven't heard that. When I talk about the lack of actionable science, in my mind, it's around those causes of preterm birth per se. In other words, what intervention would you give to that pregnant person where preterm birth is threatened that would potentially extend that gestation? I have not heard, I mean, we heard microbiome yesterday and some interesting science around that, but I get the impression, at least from listening to Anjali and other experts in this area, that we don't know. We don't have actionable science to where we can say, well, here's an intervention worth trying. I mean, for whatever, 15, 20 years, yeah, we tried progesterone and, you know, maybe tocolytics, but beyond that, we don't have any understanding. But again, I'm just listening to what others are saying and giving it back. Anjali is more of an expert than I am on this point.
Then please, Anjali, I'll have you go next and then Mark. Oh, sorry, I think you're on mute.
Yeah, I am. I got it. I just want to clarify. So the focus of my research is not preterm birth. And I would definitely, there are lots of other people on the panel who are the ones that are most equipped to answer the questions about where the state of the field stands. I think two things I just want to highlight is in terms of what's the information that we need. I think what we know as MFMs is that we see lots of people who come in with cervical shortening or premature dilation or contractions or ruptured membranes who do deliver and who don't deliver. So I think the first thing we need to do is be sure that we're investing in collecting information about people with all of those different presentations so we gain a better understanding of what is the difference between people who do go on to deliver early or not. I think some of what is the current conversation around betamethasone has to do with, you know, what are the patients that actually come in and we have this concern for preterm birth and it ultimately doesn't happen. We all know that we would love to be able to target when we're giving our interventions like betamethasone to be able to make sure that we're getting the best outcomes that we can. And at this point, we still don't really have enough information to be able to do that effectively. How do we know who's going to deliver more than 48 hours, less than 7 days away? So that information comes together. I think the other part of it is that while you said, you know, maybe all 24-weekers are relatively similar, but some of the other things that we talked about is, you know, whether the mom also has had, the pregnant person has some pathophysiology going on that's impacting her, how does that impact outcome? And then also in terms of our inflammatory aspect to things versus placental etiologies, you know, how do we understand how all those things come together? So I think, again, that idea of investing in upfront collection of information as people are presenting with some of the things we know are potentially putting people at risk for preterm delivery, and then also for those who do deliver preterm, collecting as much clinical, biochemical, placental pathology, all the pieces of information we can so that we're able to really be able to think about what these targets are and to investigate further. And then I think just the last thing I would say is that, again, I don't want to hold us to a different standard here than we do in the rest of medicine. I think there are other fields where, though the information is incomplete, they continue to invest in all of these different lines of investigation. So what I hope no one walks away from this conversation with is the idea that we're just stuck and we can't do anything until we advance the field in this very significant way about understanding, you know, all of the phenotyping. I think that, and like I said, the people who have focused their research and their research careers, of which there are several on this call, can speak to this better. I think the idea is just that we can't leave those lines of investigation until we've really felt like we've been able to answer those questions more effectively, as we're also considering what are the different therapeutic approaches we want to offer.
Very, very important comments. Thank you, Anjali. Mark and then Anne.
Yeah, I'll sort of echo many of Anjali's comments, but I think it is key and would be very helpful to be able to have some sort of a biomarker or something that would more accurately tell us, you know, when a woman is at risk for delivering preterm, especially significantly preterm. We don't have that. To Skip's point, that I'm not aware that there are any studies out there that have looked at outcome differences in the very preterm babies as a function of maternal situation. I would speculate that, and probably worth doing actually, would speculate that mothers who present with chorioamnionitis, the babies might have worse outcomes. But, you know, if you compare a mother who is, you know, traditional cervical incompetence versus, you know, spontaneous labor without evidence of infection, versus a mother who gets section for preeclampsia, I'm not sure there's data that says those outcomes in those babies are different.
Thank you, Mark. Very helpful. Anne, please.
Yes, I was going to make a similar comment. I think the data about chorion is maybe the most robust, and all the other etiologies we have definitely a gap. And I also wanted to react to some of the comments made about accepting risk, and Christy's comment about maybe if we had something that was going to reduce the extreme preterm births, we might accept more of that uncertainty, because we know that those infants are at highest risk for the most severe outcomes. So I think it's important to kind of highlight that we're having a very general conversation about outcomes for a very heterogeneous area. And if we had more information on the front end about the etiology, as to Skip's point about different subsets or different populations or different targets, then we may have more ability to accept some of the uncertainty around these outcomes. And we heard from families yesterday and in other patient listening sessions that we've had about investigational therapies and outcomes. The acceptance of risk, it really goes down to that benefit risk. So if we can avoid a few extra days of hospitalization or some of these more milder outcomes associated with the typically later gestation preterm births, with a very low risk therapy or something that's very targeted and has good evidence behind it, that may be something acceptable. And then that outcome discussion is very different than something, with an enriched population, to Tamara's point of, a group that you can select that has a very high risk of extreme preterm birth. And then the outcome discussion is, I think, different there. So I think, some of this, we want to have some general concepts, but at the same time, I think we have to keep in mind that, some of these things may be very specific and specific risk benefit discussions when it comes to a specific product.
Thank you, Anne. Skip, please.
Yeah, Lynn, I'm looking at some of the comments, and I'm tempted to just give a few quick answers from my perspective on some of the comments. One I answered in written form, which was the question about the neonatal follow-up, and what I said was, Johnson & Johnson is doing some research in maternal fetal indications. It's public knowledge in hemolytic disease of the fetus and newborn, and fetal and neonatal autoimmune thrombocytopenia. And in those, we're being required to do 2-year follow-up. And I think Anne had mentioned 2 years. So that seems to be the current standard at this point, with recognition that if you required 5 years and longer. If you asked a neonatologist, I was taught you really don't know your outcome until you get to about 8 years of age, because even at 4 or 5 years of age, things like Bailey is not a great predictor of school performance, but you have to have a realistic point. And about placebo, I think I'd be curious, Anjali, comments on that. I mean, it goes a little bit to that, what can we offer, and that angst about, when all you can have offer is a placebo-controlled trial, how does the placebo group feel? But we are conducting placebo-controlled trials, and in the right circumstances, and in the right locations, and so it's not impossible to do that. And arguably, what you could say that that optimizes the ability to see a treatment effect, and that's the honest thing to do when you don't have anything else to offer. And then, there's a question about real world evidence. I mean, I think, yeah, that can form a part of the packet, but I certainly don't see, I don't see real world evidence replacing the need at this point for some form of a controlled clinical trial when we're talking about a field where there's really nothing. It's a little hard to imagine what kind of real world data might be generated then that could potentially help. Maybe as a control group, that's a separate issue. But anyway, I just saw a few of those comments, and I was tempted to sort of, I realize we don't have a lot of time to delve into each of these things, but just thought I would at least mention a few of that, at least give my thoughts on.
Thank you, Skip. I will have to circle back because I will have failed as my job as a moderator if I don't go back and specifically ask, what do you think the endpoints ought to be right now based on the state of the art? But before I ask that question, I'll have you guys think about it. Before I ask that, I'm going to go to Tamara and then Anjali again.
Well, Lynn, that actually speaks to my question for the panel. I wanted to delve a little bit more into this tension between simple, easy to collect clinical trial endpoints and meaningful, patient-centered, nuanced endpoints. And I understand we have to operate within current regulatory framework and that you have to have traditional power calculations and things that you can quantify with some degree of certainty. All that being said, I think there is room for us to improve on the way we're thinking about current clinical trial endpoints for these preterm birth trials. And so, I try to put on my hat as a neonatologist. What's important to me in the family when a baby's born at 25 weeks is very important to what's important to me in the family when a baby's born at 30 weeks versus 34 weeks. Each of those gestational ages have important NICU milestones that are unique to each gestational age bucket. They are quantifiable. We will have to have a bit of a nuanced outcome. I like Dr. Hudak's idea of distributions of these things in the different gestational age buckets. Statistically, it's definitely possible to compare groups. But I try to put on, the hat of somebody in a pharmaceutical company who is looking at these nuanced outcomes that we're suggesting and saying, this seems, I'm just thinking of the number of data queries, So, right now we've gone to this very reductionist approach. Do they have RDS? Yes, no. Do they have this? Yes, no. But it doesn't capture the nuance of what's important. And I also think it doesn't allow us to see potential benefit because we've oversimplified these clinical trial outcomes. So I'm just really curious to hear other people on the panel's thoughts about that tension.
Yes, thank you, Tamara. I feel that tension too, almost every day of my life at FDA. So Anjali, please.
So just to start there for a moment, and then I just want to make one comment about the prior discussion. So I think one thing we haven't really talked about here is the thing that we deal with every day, which is our limited resources, right? And trying to understand, like, within the context of those limited resources, how do we decide where we're going to invest that? And I think some of that goes to kind of this idea overall, because when we want to ask for the resources to be able to follow patients, follow a groups of patients that may differ, who may have different barriers to enrollment, who may have different barriers to long-term follow-up. How do we think about how to prioritize within that to a certain extent? And I agree with you. I think it's a tension that we're probably not going to resolve, much as I would love for us to be able to. But I think it's more to say that one of the other things that's important is a robust discussion with representation from pediatricians and obstetricians to understand the way in which these particular tensions play out in our populations that is different from the general adult population. I think here with this group, I'm preaching to the choir, right? Like, everybody knows those things, but I think it's an important discussion to be able to take beyond our own groups of researchers so that everyone understands why there is sometimes some more investment that's required when we're thinking about all of these different things, and also to understand what the particular barriers are to research in our populations and why it's important to invest in that in particular. Just for one moment, I just want to return to the idea of kind of real world data and where that comes in. I do think that with the advent of the electronic health record, with more robust administrative data sets, and with some of the more advanced statistical methods, including quasi-experimental methods, there are some opportunities to be able to collect some information that can inform clinical trials. I think at this point, it is hard to completely say that, those can replace a clinical trial, but I would say that that's the place where we can gain more information from the experience we already have, is by applying some of those methods to the data that's been collected. I think one thing that's very important to think about in that context as well is the fact that so many deliveries in the United States are covered by Medicaid, and often our administrative data sets that include Medicaid participants are not as robust as those that are privately insured. And so again, in sort of thinking about where are those gaps, where if we had more information, we would have a better ability to really understand what are all of these different factors that contribute to both the onset of preterm labor and eventual spontaneous preterm birth, and also the ways in which people might be able to be compliant with a recommended treatment, and also how that treatment might have demonstrated efficacy. I think we want to make sure that we are collecting more robust information about Medicaid beneficiaries, because those are a lot of who our obstetric patients are.
Thank you. Thank you, Anjali. So I know Ann and Skip, you both have your hands up, but if I don't ask this question, I only have 9 minutes left, and I really want to get an answer from each of our panelists. So I heard, look, well, we've heard all through the last 2 days, gestational age alone, probably not the best endpoint. Binary composite, right, where you have a whole bunch of things, but your answer, did you have it or not, probably not the best endpoint. But what if I could get from each of you an idea about what an endpoint would look like, or what endpoints could be feasible, or what additional information do we need before we consider those endpoints? I think Anjali gave us some, and Skip gave us some already too, right? We need to get everybody together. We need to hear what's important. We need to make sure it's measurable, and we need to make sure that it's able to, that all patients who are involved in the databases we use, Skip, to your point about bringing together, they got to be representative. So it can't be just a proprietary EHR. We got to look at, who's delivering these babies, to look at those endpoints that we could potentially model. But I want to hear from each of you about a potential strategy to get to an endpoint that could be considered in prevention of spontaneous preterm birth. And Skip, I'll start with you then, Ann, because you both have your hands up already.
I'll just make a couple of comments. I was taught, I hope that this is still true, that continuous variables are much more powerful than categorical variables, that you lose data when you take a continuous variable and you convert it into a categorical variable. So I would encourage us to somehow think about our neonatal outcomes along a continuum. And I think it was maybe Mark who brought this up around, instead of just treating them all equally, weight them. And I would argue that it's an important place of parents to weight the significance of these morbidities and the impact of that on the quality of life of that infant, and of course their quality of life. Now that would be very different in my mind than trying to put a parent-reported outcome together, because we all know that the construction and validation of all those is a big deal. But if we decide that we want to weight a certain morbidity at a certain level compared to another, which is more impactful, I think we could maybe begin to develop a continuous variable. And I again would say we should model that. We shouldn't just make it up. Model what that looks like using existing neonatal data to say, if we decide to trial with this endpoint, what would be the difference on this endpoint between a 24-weeker and a 28-weeker? Again, assuming no difference in utero environment. I mean, that may be, but we could at least model it and say we could, if we saw a 4-week difference in gestational age, or a 3-week difference, or a 2-week difference, we might be able to see a difference in that neonatal outcome with what sample size? Because that's the issue. Are these trials going to be feasible? And if we discover that that outcome means we need thousands and thousands of babies, and we need them all to be in the 24 to 26 week range, that's going to be tough.
Thank you, Skip. Ann, please.
Yeah, Lynn, so to address your question about where we are today and where we want to be, I think, today we, maybe have some of these more complex composites, I'll say, that maybe give us mild, moderate, severe, or some gradation of these outcomes into buckets. But the path to either a continuous or even some ordinal scale, like the global rank scale that I mentioned, is definitely a work in progress and something that we just don't have available to us right now. And to Tamara's point, the word nuance makes me very something I want to capture as a clinician, and as a regulator it scares me because I think there's some subjectivity there, and some are we able to interpret those types of outcomes, which is why we go back to that simplistic like black and white yes no. So I think there needs to be a middle ground. I'm personally excited about the work in the global rank scale because it does what Skip is suggesting, and taking a lot of input from multi-stakeholders to try to put some weighting to these different outcomes, but still have some kind of ordinal or ranked scale that has some validity, that's not more of a subjective, how do we capture all of these nuances across all of these different morbidities that may be different in importance to different families, and those kinds of things. So I think we need that middle ground, and something like that global rank scale is something that I'm personally excited to see where that heads, because I think that may give us something like that.
Terrific. Thank you so much, Anne. Mark, please.
Yeah, so I think seems like we're coming to some sort of consensus on this. So I'd just like to, just give you some quantitative data to establish the fact that really, if you look below 30 weeks and after 30 weeks, it's vastly different. So for instance, if you look at mortality, at 24 weeks, it's about 28%. By the time you get to 26 weeks, it's about 12%. By the time you get to 28 weeks, it's about 5%. So huge differences over a short time of mortality. Other morbidities, are also, they're similar. Chronic lung disease, 70% chance when you're 24 weeks. It goes down to about a 20% chance when you're 28 weeks. So huge changes over time in some of these morbidities. So I do think that all of these things need to be examined individually, compositely weighted. And, certainly the minimum bar has to be that if you do prolonged gestation, you don't worsen your risk. I mean, you may not reduce it to, what you might expect. If you go from 26 weeks to 29 weeks, maybe you don't get that 30% reduction, maybe you get a 20% reduction. Then you have to ask, is that okay? I think it is. But on the other hand, you have to ask, if you don't see any improvement in some of these morbidities by extending gestation, then I'd be really worried about long-term outcomes being worse. Because generally, when you sort of have a profile of morbidity at a later gestational age, you're going to predictably have a worse outcome later on. So, and then for the ones that are over 30 weeks, I think the outcomes are going to have to focus on issues that, are important, like length of stay or post-menstrual age of discharge, or some measure of, family quality of mind, or whatever, however you want to phrase it, because you're not going to see these morbidities in any large concentration once you get past 30 weeks.
Terrific, Mark. Thank you. We only have a couple minutes left, so I haven't heard from Tamara and I haven't heard from Anjali, so I want to hear from both of you, and Christy, of course.
Well, I'll just say quite simply that I think my takeaway from this meeting is the great import of incorporating the patient voice in a much louder way when we design these clinical trials. I know that the logistics of that is not always the most simple, but I just think it's incredibly important for these type of obstetrical conditions in particular, and this particular entity of preterm birth, which is a extremely traumatic experience for the mother, for the family, regardless of the neonatal outcome. And so I hope moving forward that designers and sponsors of these clinical trials really bring the patient voice in very early and very loudly.
Thank you. Anjali, please.
So I think that all the comments that have come forward today, as I said, I want us to kind of combine that idea that we still have work to do with also the idea that it's not hopeless, right? I think we've seen lots of common themes. I also think that when we think about the limitations of gestational age, we also know that gestational age nonetheless is something that gives us important information, and kind of looking at things in combination there as well. I think that, we have in general done a really good job of being a community and talking about these things together. I think we've more and more understood what are the important kind of maternal outcomes and in addition to neonatal outcomes that we want to think about. And I would just say that let's continue to do that. And I think by, as Dr. Lewis said, centering the patient voice does that, right? Because they understand what is the implication for this child who's born early? What is the implication for them in terms of their life? What is the implication for the entire family? And so sort of thinking about when we're trying to have an impact, that we're thinking about all of those different elements. It's not easy, but I'm glad that this conference helps us to focus on it. And I hope there'll continue to be a robust discussion like this.
Wonderful. Terrific comments. Christy, I'll give you the last 30 seconds.
Thank you, Lynn. The first comment I'll make is that the clinical outcome of interest really is how the baby is doing, how the baby is functioning, feeling, and surviving. So these binary endpoints that we put in the composite neonatal endpoints really are approaching that level of need for clarity for us. So I'm very impressed with the patient's testimonies and the family's experience. So endpoints such as length of stay, time to extubation, those type of endpoints in my mind are clearer in terms of telling us how the babies are functioning. So that's what I'm hoping to hear more about. And I recognize that we have so much more work to do. So thank you, everybody.
Really terrific. I know we're a minute or two over, so I will turn it back, I believe, to Nancy, but I want to thank again our amazing panelists. Thank you so much for your insights. I've been madly taking notes, so I hope I am able to reasonably describe what we talked about in the session towards the end of the day. But Nancy, back over to you. Thank you again.
Great. Thank you so much, Lynn. Yes, we're going to take a break. We will come back at 2:35 for session 5. Thank you.
Welcome back, everybody. My name is Michal Elevitz. I am the Dean for Women's Health Research and the Director of the Women's Biomedical Research Institute at the Icahn School of Medicine. It's a pleasure to be here today and yesterday. I am moderating session 5 on discussion of the dose finding and trial design for preterm birth therapeutics. During our session, please feel free to use the Q&A chat box to submit your questions for the moderated discussion portion of our session. Before we start, I'd like to review the objectives of this session. The intention of the session is to help identify the data needed to support clinical trials for preterm birth therapeutics, as we've discussed for the last day and a half, and the different study populations and control groups that should or maybe be used in these trials. We will also consider how the physiological changes in pregnancy affect dose finding and drug metabolism. The session will consist of 2 15-minute presentations from experts at the FDA and will be followed by a broader panel discussion. To begin, we will first hear from Kimberly Hatfield from the US FDA. Kimberly, whenever you're ready.
Are you able to see my video? I cannot at this point.
Yes, we can see it.
Okay, you can see me. Wonderful. Thank you. Well, I want to thank the organizers today for inviting me to speak today about the non-clinical safety assessment of pharmaceutical products to be administered during pregnancy. Next slide, please. The objectives of my talk today are to explain the content and the characteristics of non-clinical studies that are conducted to predict the safety of a pharmaceutical product to a developing human in the prenatal period. And I will say right now that I have no disclosures to make. Next slide. So first off, why do we conduct non-clinical toxicity studies? Well, animal studies are actually mandated in order to evaluate drug toxicity prior to first-in-human trials. And this is put forward in the Food, Drug, and Cosmetic Act. And then the CFR is also has listings that animal toxicology is necessary to support first-in-human clinical trials. And we have developed regulations and guidance over a number of years for both how and when to conduct non-clinical studies. And these guidances are both international through ICH, and then there are also FDA guidances. And this link that is active here is a searchable database of all of the FDA guidances that you can access. But in the context of preterm birth that we've been speaking about these past 2 days, I want to highlight the importance of reproductive and developmental toxicity studies. And these are just one part of the total non-clinical assessment of a drug product. Next slide. So what exactly do reproductive and developmental toxicity studies measure? Reproductive toxicity mainly looks at the effects on the mother. So structural and functional alterations that affect reproductive competence in both sexually mature males and females. We look at male and female fertility, parturition, and lactation in this space. And then developmental toxicity primarily looks at the effects on the offspring and adverse effects on the developing organism. We look at outcomes such as mortality, alterations to growth, structural abnormalities, and functional impairment. Next slide. So how do we evaluate reproductive and developmental toxicity? Well, we have a standard non-clinical battery of assessments for a full non-clinical package, but reproductive and developmental toxicity can be evaluated in the early repeat dose toxicology studies, whereas we would evaluate a number of doses over a specific duration in either rodent or non-rodent species. And we would specifically look for any histopath evaluation of the reproductive organs and if any sort of changes are occurring versus controls. We then could look at the evaluation of the menstrual cycle, and if there's any extended time in any one stage of the estrous cycle, that could indicate some sort of hormonal perturbation by the drug product. We also have a standard battery of reproductive and developmental toxicology study designs, where we specifically look at various development stages. First, we look at fertility and early embryonic development. The second study is to focus on embryo-fetal development, and the third study is to focus on pre- and postnatal development. Next slide. So this graphic shows a little bit more of the detail on what each of these studies investigates in terms of drug exposure on the developing offspring and the mother. So first, on the right-hand side, we look at the fertility study, and this encompasses dosing of a drug product prior to mating, through mating, and then up until implantation of the embryo. The second study, embryo-fetal development, looks at drug exposure at the time, starting at the time of implantation, all the way through gestation up until parturition. While the third study, the pre- and postnatal development study, looks at the drug exposure from implantation through gestation and birth of the offspring, and then exposure of that offspring through lactation, because the mom is treated during those studies up until the time that they are weaned. And so in the preterm birth space, we are primarily focused on results from the embryo-fetal development and the pre- and postnatal development study, because the drug exposure encompasses the same types of exposure that a human subject would have. Next slide, please. So I wanted to give you a little bit more perspective as to what these non-clinical studies encompass. The embryo-fetal development study is particularly important for drugs that could be used during pregnancy. These are evaluated in two species, both a rodent and a non-rodent. And the graphic shows here that dosing occurs from implantation, which in the rat is around gestational day 6, and in the rabbit at gestational day 7. And dosing continues through the organogenesis period up until gestation day 17 or 19. And then the animals are C-sectioned and the offspring collated in order to evaluate toxicities at gestational day 21 or 29. These studies should have at least 16 pregnant females per dose group, and this really allows us to get a good amount of statistical power in evaluating our studies. The average litter size of the rabbit being 6 and the rat being 12 also gives us a large number of offspring to evaluate. And this helps us to be assured that any findings that we see are increased above the natural background and above control levels. Next slide. So the endpoints assessed during the embryo-fetal development study are mainly the maternal endpoints. We look at body weight changes, evaluation of general toxicity to organ systems. And then we also look at embryo-fetal development and pregnancy parameters, focusing on gravid uterine weight, alterations in the number of fetuses, any embryo-fetal loss, either pre or post-implantation, and then effects on the placenta. And then we also look at fetal health endpoints, such as altered growth, body weight, and then a gross evaluation of external visceral and skeletal development. Next slide. The pre and postnatal development study is also important for drugs that could be used during pregnancy or for a pregnancy-specific indication, or also for those administered during lactation. We only evaluate one species in these studies, which is usually a rat, and the dosing, as I mentioned before, occurs from implantation through the end of lactation. So in this graphic, we see that one dosing starts around gestational day 6, continues in the mother through gestation, the mother is allowed to deliver, and then exposure to the pups continues through lactation via the mom. At postnatal day 21, the pups are weaned and some evaluations are done. And then one male and one female offspring per litter are selected for rearing to adulthood and mating to evaluate any drug effects on fertility or reproductive competence. And again, the number of animals in this study is suggested to be about 16 pregnant females per dose group. Next slide. So the pre and postnatal development endpoints assessed are different than the embryo-fetal development study, mainly because the offspring are allowed to mature while on study, and the mothers are not sacrificed. So we look at endpoints such as maternal health and maintenance of pregnancy in a broad sense, such as duration of pregnancy, whether the whole litter is lost, or any live or dead offspring at birth. But the offspring that are exposed in utero or via lactation, we really take a close look at body weight, any pre or post-weaning survival and growth, sexual maturation, fertility changes, physical development, sensory function reflexes, and then also behavior, learning, and memory. And then the F1 generation that is allowed to mate and their offspring are not exposed directly to the drug product, but we evaluate them by an external exam just to see if there's any sort of effects that could happen generationally. Next slide. So I want to shift a little bit into the timing of non-clinical studies and when non-clinical studies are typically requested and conducted during a pharmaceutical development program. The graphic here shows kind of the drug development program starting from the left at discovery, and then once we enter first in human trials at phase 1, 2, 3, and then through the marketing application. So before first in human trials, the applicants usually outline a number of non-clinical studies for us where they've outlined mechanism of action, receptor and organ targets. They have conducted some pharmacokinetic studies in animals to look at exposure in circulation. They've also conducted some general toxicity studies in non-pregnant animals to determine any target organs, safe doses, and then they're also conducting genotoxicity studies to evaluate that. Next slide. So a typical development program for any indication that would use a general population, such as males, postmenopausal individuals, or individuals of childbearing potential with pregnancy testing and contraceptive use, is kind of shown here. And the general toxicity studies with the reproductive organ assessments are conducted prior to phase 1 and kind of during phase 1 in early development. The reproductive studies, however, are conducted around phase 2, phase 3, and then the PPND studies are conducted usually in parallel with phase 3, with the results submitted at the time of the marketing application. Next slide. However, for a clinical trial for a pregnancy-specific indication, or a clinical trial that specifically enrolls pregnant individuals, all of the reproductive studies, the FEED, the EFD, the PPND, also the general toxicity and genotoxicity, need to be complete before pregnant individuals are enrolled. And this is for us to adequately assess the risk to both maternal risk and fetal risk of the drug product before anybody is exposed. So in these types of trials, generally phase 1 enrolls non-pregnant subjects to get an idea of some pharmacokinetics, dose selection. And then phase 2 and 3 generally enrolls the pregnant subjects. So in these cases, all of their reproductive study development needs to happen before phase 2. Next slide. So we extrapolate the animal findings to humans by collating all of the information from these studies, the repeat dose, EFD, PPND, and they're going to provide a dose to us that produces no toxicity in animals. And this is considered the no adverse effect level, or the NOEL. This NOEL is then compared to the therapeutic dose in humans, and we generate a multiple of exposure. So this multiple, for example, 10x, tells us that at 10 times the therapeutic dose in humans, there is no toxicity in the animals. So the larger this multiple of exposure is, the safer the drug is considered for the human population. And this multiple is incorporated into the assessment to guide dosing decisions and communicate risk. Next slide, please. So the animal data really does help to inform human risk, especially when there's very limited or no human data available, especially with drug products that are novel. Negative animal data can support further clinical testing, and even positive animal data can really help to define a safe human dose range. And again, we look at the multiples of exposure to determine if there's more or less risk going on. There are some limitations because animal data could indicate a non-monitorable toxicity and potential harm to the fetus that we just can't keep an eye on during dosing. Also, it could indicate the risk benefit is unfavorable. Next slide. So all in all, this goes towards a huge decision process that we as regulators don't take lightly. We have a review team that encompasses a number of different disciplines, and we integrate all of the information from both non-clinical and clinical studies to determine whether or not a drug product is safe to use in a pregnant population. We ask ourselves questions like, are the animal findings relevant to humans? Can we estimate the fetal drug exposure? How much is compared to the mother? Which trimesters are involved? Could the fetal brain be exposed? Could adverse effects occur in the fetus more so than the mother? And then we incorporate all of this data to really make a decision because it comes down to benefit versus risk. How serious is the indication for the mother? How serious for the fetus? Is the benefit certain? Is the population well defined? If the drug is toxic to the fetus, is it monitorable or reversible? And if the mother is not treated, is there a risk to the fetus? So we take into account all of these questions when we're making our decisions on whether trials are safe to proceed. So I want to conclude that the reproductive and developmental toxicity studies in animals really do provide essential information for the clinician to make a treatment decision and to inform the patient of risk, especially in this preterm birth space and dosing of pregnant individuals. Next slide. This is just a reference slide to help you and provide guidance for future non-clinical development towards any programs. And I'll be happy to answer questions when we have the panel discussion.
Thank you, Kimberly, for that really thorough, excellent presentation. Our next presentation will be from Donnie Tran from the US FDA. Danny, whenever you're ready.
Hi, thank you very much for the kind introductions. And I would like to thank the organizer for inviting me to discuss at this very important meeting. My name is Donnie Tran. I'm a clinical pharmacologist, and I'm Deputy Director of the Division of Cardiometabolic and Endocrine Pharmacology. Today, I would discuss at a high level the clinical pharmacology program would focus on dose-finding consideration for prevention of preterm birth. Next slide, please. This is a disclaimer, standard disclaimer. The view and opinion presented today are mine and should not necessarily be that of the FDA. And I have no disclosure to point out. Next slide, please. So the outline of my talk is as follows. First, I'll go over a very brief introduction to the 3 phases of drug development, as Dr. Hatfield just put it out, phase 1 to 3 in clinical. I also go over the general recommendation on dose-finding studies, as well as the effect of pregnancy on pharmacokinetics or PK. And lastly, I'll discuss the potential implication on dose finding for prevention of preterm birth. Next slide, please. So in terms of the general drug development phases, you've seen in the last presentation, for clinical phase, that's phase 1, 2, and 3 drug development. In phase 1, we typically have single ascending dose and multiple ascending dose studies. And this is to assess pharmacokinetics, safety, and tolerability of the drug. We typically also have studies to evaluate the effect of intrinsics, such as hepatic and renal impairment, extrinsic factors such as food effect, and drug-drug interactions. These studies would help basically tell us information on whether any of these factors could impact the PK, and therefore a dose adjustment may be needed in the phase 3 trial later on. Now, for a program where repurposing drugs of a previously approved drug may be used, in this case, we may rely on clinical pharmacology findings from the prior studies as appropriate. However, it should be pointed out that often pharmacokinetic in pregnancy is not available, and in this case, it would need to be evaluated when we plan to use it in pregnant people. In phase 2 studies, usually this is when we study in the target patient population. First, usually you have a proof of concept studies to see if the drug has a chance at efficacy. And then subsequent to that, there should be a dose finding study. This dose finding study is important to find the optimal dose to study and confirm in the phase 3 trial. And lastly, in the phase 3, we have a large clinical trial to assess the safety and efficacy of the proposed dose. Next slide, please. So in terms of dose finding studies, typically we recommend that sponsors should conduct a phase 2 dose finding study to help them select doses for phase 3. This is really important because identifying the most optimal dose to take into phase 3 is critical to ensure we have the best chance of getting a product that have a good risk-benefit profile. Is too high of a dose can lead to unacceptable toxicity, and too low of a dose may risk not showing any efficacy or not adequate efficacy in the phase 3. So it's really important to do this. In any case, the doses that selected for further study in phase 3 should be well supported by robust scientific justifications. Next slide, please. So this slide shows some dose-finding studies considerations for phase 2. So in phase 2 dose-finding study, we recommend that should include a range of doses to allow analysis of dose response and or exposure response relationship. In this case, we recommend at least 2 dose level in addition to placebo control. And placebo is helpful to determine that the effect that we observe in the treatment arms are due to the drug and not due to some other reasons. And the dose levels chosen for these phase 2 dose finding study should have adequate separation to avoid the risk of being at the plateau of the exposure response relationship. When the doses chosen close together and they're too high, we may see a response, but there's potential for a lower dose that would achieve the same amount of efficacy, but at a lower risk. Similarly, on the other end, if you have exposure doses that are chosen that are close together, but at the low end of the exposure response relationship, we may be able to increase that dose and achieve a much better efficacy while not increasing the safety concern. So because of that, we really recommend that there should be good dose separation to help assess that during the phase 2 study. Also, for drug with high PK variability, that a greater spread of doses should be considered so that we can see a separation in effect of the doses that are being tested in the phase 2 trial. Also, a wide range of dose may be needed in cases where there's not a good pharmacologic endpoint or biomarker that provide us with guidance on what doses may be needed. So in this case, because we don't really know what the target concentration may be needed, so you may need to study a wide range of doses to ensure that at least 1 or 2 of the doses being tested would have efficacy. And overall, we recommend that after the sponsor have determined what their proposed study design and doses, that they should come in and talk with the FDA so that we can review and provide our recommendations. Our goal is to help sponsor develop the best products to ensure that there's a good risk-benefit profile and that the study is designed in a way that would be helpful in making that determinations. And also another general recommendation is that the sponsor should use modern simulations to help predict the doses and the dosing interval that would provide the best risk-benefit balance to help them ensure that the phase 3 trial would be adequately have a success rate that will be useful. Next slide, please. So the typical dose-finding study design are shown on this slide. We have parallel study, crossover study, and titration study. These are the typical ones that I see. So in the parallel study design, the subjects are randomized to several different fixed dose groups, and the placebo inclusion is desired unless it's not ethical, of course. And this trial design allow us to assess a variety of disease conditions, including those that have a long, require a long time to see an effect. And then because of that, this is most likely the case that will be applicable to the prevention of preterm birth, because most likely the endpoint that we're looking at is either at gestational age at delivery or some sort of neonatal outcomes, which will require dosing to delivery. Another study design is the crossover study. In this case, subjects are randomized to a crossover of several different dose levels. This study design is good when there's rapid drug effects, that's rapid onset as well as rapid return to baseline. This would allow the same subject to be given different dose level, and we can see the outcome between each treatment interval. This may not be applicable for prevention of preterm birth unless, in the future, we can come up with a more rapid responding biomarker that can be identified. A third study design is the titration study design. In this case, it could be forced or titrated. In a forced titration study, the subject is titrated from a low to high level, high dose levels in a sequential manner, regardless of the response. In the titrated to response, they would be titrated up, but they'll stop at a given response is achieved. In this study design, it's one of the drawbacks is that it cannot distinguish the response due to the increasing dose with each titration versus the response due to the increase in time on therapy. Because as you increase the dose level, the patient would also have been on longer duration of therapy. So it's hard to, with this study design, we cannot distinguish between those two. Next slide, please. In considering the effect of pregnancy on PK, we know that from the literature that there are pregnancy lead to different physiologic changes to the body. And this may lead to changes in pharmacokinetic of the drug. For example, oral absorption may change due to the presence of nausea and vomiting, increased gastric pH, prolonged gastric emptying, and prolonged small bowel transit time in pregnant people. Distribution may also change due to increase in plasma volume, increased body weight, increased body fat, or decreased plasma albumin, etc. And metabolism and excretion may change due to changes in activity of metabolizing enzymes. Some metabolizing enzyme activity are decreased during pregnancy, and others are increased. So it depends on the drug of interest. This may cause an increase or decrease in the drug exposure. Metabolism and excretion can also change due to increase in hepatic blood flow that's seen in pregnancy, increased renal blood flow, and increased glomerular filtration rate. So in short, at a high level, pharmacokinetic may be affected in pregnancy, and the impact may be seen in the first trimester and with larger impact in later trimesters. Next slide, please. So it's noted in the previous slide where pregnancy may affect the PK of the drug. Characterizing PK in pregnancy is really important. However, we should know that the pharmacokinetic and the safety of a drug should be characterized in non-pregnant subjects before proceeding to dosing in pregnant subjects. This is to minimize the risk that in the mother as well as the fetus. So it is important that we have this information to help determine what exposure may be seen in pregnant women. And in that case, we can determine what dose we might want to take into the pregnant people. Also, as our target patient population is pregnant people, pharmacokinetics should be characterized for the periods in which the drug would be administered. For example, the first, second, and third trimester. I would suspect most of the therapy would be hopefully be administered in all trimesters so that the patients would be carrying out to the third trimester of pregnancy. So in this case, we would be expecting to get PK in all trimester of pregnancy. Now, pharmacokinetic can be collected in a clinical study of pregnant people being treated for the prevention of preterm birth. However, a separate PK study could also be conducted in pregnant individuals if the risk to the patient and the fetus is minimal. Next slide, please. So there are some considerations that are unique to dose finding for the study design for the prevention of preterm birth. And I outlined some of them here. As noted previously, drug dosing would likely span all 3 trimesters of pregnancy. And we know that, in the physiologic changes in pregnancy may lead to changes in PK. So therefore, the dose may need to be adjusted during pregnancy as it progresses to maintain the target drug concentration that we might want to achieve. Nothing with the dose finding for prevention of preterm birth is that it's likely that we'll be dosing the patient throughout the 3 trimester of pregnancy. So this would also be a good opportunity for a longitudinal study design where PK and maybe pharmacodynamic dynamic endpoint as well can be captured in the same individual. So this way we will have the cleanest way to understand the impact of pregnancy on PK and PD in an individual. As mentioned previously, most likely a parallel study design would likely be needed if the endpoint is delivery or some neonatal outcomes. Currently, it's not clear if there are other pharmacodynamic endpoints that can show rapid response, rapid onset, and rapid return to baseline that could be used to predict clinical efficacy. However, if in the future these endpoints become available, then other study design may also be applicable. Next slide, please. So in summary, the dose response or concentration response information is really critical to select an optimal dose for phase 3. And we recommend a dose finding study be conducted to obtain this information to help guide dose selection in the phase 3 trial. As noted before, pharmacokinetic may be altered during pregnancy, and this should be considered, and dose adjustment may be needed as pregnancy progresses if a particular drug is impacted by PK during pregnancy. And that the parallel study design is the most likely study design for a dose finding study in prevention of pregnancy, prevention of preterm birth. However, in the future, if there are other endpoint that has rapid onset and rapid return to baseline, other study design may be feasible as well. And that concludes my presentations, and I look forward to the panel discussions.
Thank you so much for that very interesting and for those of us who do this research, concerning thought that we'll get to hopefully in the discussion. I'd like to invite our additional panelists to join us for discussion about the content covered in this discussion. I would like to remind everyone attending the webinar that full bios are available, so I'm not going to introduce everyone now, but just please realize the great expertise and really esteemed people that we have joining the talk. So Kevin, Steve, and Kelly, are you all on? I'd like the panelists to introduce themselves and give us a quick kind of opening remarks, if you will, and maybe to speak a little, not just about this talk, but from your lens and kind of summing up what you think this discussion should actually, the high points of this discussion. So Kevin, I'm going to start with you.
Great, thank you very much, and good afternoon to everyone. First of all, I'd like to thank the meeting organizers for inviting me to this meeting, as well as the audience for attending. A lot of good information is being shared, and I hope it's useful for all people involved. As my bio will tell you, I'm the Associate Director for Clinical Policy and a Senior Bioethicist in the Office of Clinical Policy, located in the Office of the Commissioner. My office is responsible for developing cross-center policies related to human subject protections and good clinical practice. In addition, we provide a bioethics consultative service to the review divisions in all the centers for research involving adult volunteers. This last activity sometimes involves providing an ethics consult on research involving pregnant people, should the review division have concerns about the ethics of a proposed protocol or a marketing application. FDA is committed to improving the diversity of individuals participating in research and fully supports the inclusion of pregnant women and lactating women in clinical research. Over the last 20 years, there has been a tremendous success in increasing the number of women participating in clinical research, but there has been very little gain seen in the inclusion of pregnant women in research for some of the reasons discussed today and yesterday. This is not just about numbers. This is obviously a public health issue, and there's a critical need for properly conducted clinical trials in pregnant and lactating women's people to assist healthcare providers in addressing the healthcare needs of these individuals. The gap in knowledge is huge, and it needs to be addressed. Concerns about the benefit to risk assessment in this population are real and should not be underestimated. Hopefully, the non-clinical studies that were outlined earlier and perhaps limited human experience, if any, will provide sufficient information to justify moving forward with human trials. With careful consideration of trial design, good risk mitigation efforts, and the enrollment of well-informed individuals, this type of research can be done ethically. And the final point I'd like to make is, as mentioned by Dr. Lewis earlier today, that the inclusion of the patient voice in all of this would be very, very helpful and should be considered. I look forward to the panel discussion. Thank you.
Steve, thank you. Kevin, Steve.
Hi, Michal. I'm very happy to participate in this conference and have a few slides to discuss. I was asked to discuss why Makina failed. But let me start out by saying that not much has changed in this field since we were using intravenous alcohol in the 1960s. We tried multitude of treatments in the last 5-6 decades. And it seems like there's a high level of enthusiasm for treatment, and tincture of time destroys that enthusiasm. So this conference is very timely because we need something new. I was asked to comment again on why I thought McKenna failed. And my title tells you why. I think the dosing was suboptimal. I'd like to acknowledge my collaborators in this research, particularly Dr. Venkataramanan, who was the first to develop an assay for 17-hydroxyprogesterone caprate and perform the assays on all these studies that I'm going to review with you that led us to believe that the dosing was suboptimal. I'll let the cat out of the bag and tell you the two dosing errors that we've identified. First, we think the dose was too low. Second, a loading dose was not given, and it should have been given because of the drug's half-life. Next slide. We use data from 3 prospective trials, one from the Maternal Fetal Medicine Unit's omega-3 study that described a relationship between 17-OHPC and spontaneous preterm birth, one from the OPRU, the Obstetrical Fetal Pharmacology Network, that looked at the pharmacology of this drug and its placental transport, and finally from the Obstetrical Fetal Pharmacology Research Centers, the OPRC, which also looked at the relationship between drug concentration and preterm birth. Next slide. These are data from the omega-3 study, which again was published roughly 10 years ago. And in a cohort of over 300 women, a blood was drawn at 25 to 28 weeks for measurement of 17-OHPC. This chart shows the relationship between drug concentration and preterm birth. And you can see there is a concentration relationship response. Those women with the lowest concentration, they were in the lowest quartile, had a spontaneous preterm birth of 43% compared to 25% with those in quartiles 2, 3, and 4. As this Kaplan-Meier curve shows, those women in the lowest quartile delivered much earlier than those women in quartiles 2, 3, and 4. The drug concentration in the first quartile was about 6.3 nanograms per ml. Next slide. We took these same data and fit them to using a linear regression model, fitted them, and this slide shows a statistically significant relationship between the probability of preterm birth and the log concentration of 17-OHPC. These are the log numbers at the bottom, and right above them is the anti-log, which is the actual drug concentration. We wondered if there was a particular breakpoint that could be used as a target concentration. Next slide. So in that same omega-3 cohort, we looked at these various concentrations. And again, these were concentrations obtained at the 25 to 28 week blood draw. We assumed the concentrations would be at steady state, but we know now that they were not. This 8 nanograms probably translates to a steady state concentration of about 9 nanograms. And you can see there was a significant relationship whether we define preterm birth as 37, 35, or 33 weeks. All 3 epochs were significant when we use a cutoff of 8 nanograms per ml, again, at 25 to 28 weeks, which is equivalent to 9. We'll show you later, nanograms per ml. So those studies, we think, show that the dose of 17-OHPC or McKenna was too low for about a quarter of the women. Next slide addresses the issue of a loading dose. First, pay attention to this lower right-hand side that looks at the half-life of 17-OHPC. These data were based on the OPRU study, where in 61 women, we did intensive PK sampling in the second and third trimester. And in a smaller group, we drew bloods an additional month after the drug was stopped. That enabled us to estimate half-life. And you can see that the median half-life was 14 days, and the interquartile range was 11.5 to 17.2 days. Now, these curves here are simulated data obtained from this OPRU study, where again, we had lots of concentration data, so we could develop a model that simulates trough plasma concentrations. For those that aren't familiar, trough is the time just before the next dose. So these are the trough concentrations among those women who had a median half-life of 14 days, the shorter half-life of 11.5, or the longer half-life of 17.2. Notice the numbers to the right indicate what the steady state concentration would be. Notice that those with the shortest half-life and the most rapid elimination of the drug never achieve the 9 nanograms per ml. They are always subtherapeutic, and that's about 25% of the population. Notice also how long it takes to get to steady state. For the median half-life, it takes about 70 days, and that's based on the requirement that 4 to 5 half-lives are needed to achieve steady state. So if the half-life is 14 days, if we multiply that by 5, that's 70 days required to reach to reach steady state. So for the median value, it's right here. So you can see at that point, the plasma concentrations aren't changing very much. So we've reached steady state. So for drugs that have a long half-life, such as warfarin, digitalis, and phenytoin, loading doses are given, but that wasn't done for Makena. Next slide. In this slide, again, we use our predictive model to demonstrate how long it would take to get to the target concentration of 9 nanograms per ml. This lower curve represents the current dose that was recommended, 250 milligrams weekly without a load. And you can see it takes 1, 2, 3, 4, 5, 6 injections to get to the 9 nanogram target. Now recall again, this is with a median half-life. Those with a short half-life will never achieve this 9. They will go up here and flatten out. So what strategies exist for trying to achieve this concentration quicker? Well, you give more drug initially. Well, the drug is now concentrated at 250 milligrams per ml. We can't go much higher than that because the drug will precipitate. We did a study in the OPRU where we randomized some women to 250 and some to 500 milligrams weekly. Those who received 500 milligrams weekly had no more side effects, and neonatal outcome was no different than it was in the 250. So in these examples, I'm using just the 250 milligram concentration and using 2 mLs of it at each injection, which is what we did in the OPRC study. If we just gave the first injection at 500 milligrams, you see that we reach this magic concentration after 1, 2, 3, 4 doses. If we give 2 500 milligram injections, we reach that concentration after 1, 2, before we even reach 3. Now, you could give higher concentrations. These are just examples. You could give 500 milligrams the first 3 shots and you'd get there quicker. So basically, we think these are the 2 reasons, or 2 of the reasons why Makena failed. Now, even with these pharmacologic missteps fixed, we still don't know that that would improve pregnancy outcome or the drug's effectiveness. All we are showing is that the dosing regimen was not optimal. If you can go back to the first slide. One more. No, the very first one, that one. The other thing I want to emphasize is lessons for future pharmacotherapy. How did this happen? We hear this, though, over and over again with other medications used in pregnancy. Many of the medications are used off-label, and therefore, I'm sure Dr. Tran will roll over in his grave and say that's not right because PK, PD studies are not done. A lot of drugs that are used even on label, the PK, PD studies aren't done. So we need to focus on proper pharmacotherapy for pregnancy. Thank you.
Thank you, Steve. Appreciate that deep dive into 17-OHP levels. Just a reminder for our discussion and for the group that we're going to try and stay away from talking about the McKenna trials. We can talk about why failures and how to prevent them forward, but just to keep that in mind. I'd like now for Dr. Kelly Moley to please introduce herself and give us opening statements.
You're on mute.
Still muted, Kelly.
Is that better? Thank you. Sorry about that. Thank you for inviting me to the organizers of the meeting, and it's great to see you again, Mikhail. So yes, as Mikhail said, I've been asked to speak for 2 to 3 minutes about my experience in academia, in philanthropy, in foundation work, and now in pharma, from the perspective of drug development. And I'd like to start by saying, for 30 years, I was in your shoes as an academician. I even had a patent for several products. Granted, I was an OBGYN in reproductive endocrinology, but I was frustrated that there weren't drugs that were out there for the diseases that we saw, like endometriosis and polycystic ovary syndrome. And that's one of the reasons I left. But what I've learned is that it's a very small part of the journey, that discovery, that publication. But innovation really requires discovery science partnered with pharma, because pharma is to carry the product over the finish line to a marketed product, which can require between $500 million to $1 to $3 billion. And that's not something that NIH can fund. And in the end, pharma needs to pay off the cost to finance the clinical trial costs. So for them, success depends on many things: the final product market value, what payers will pay, perceived unmet needs by the payers, willingness of pharma to finance and develop and market, and consumer conceived need for the product. Now, as we've all heard, there have been very few innovations in this space in the last 30 years. There's only been 2 drugs, and they were both recalled from the market. Poor funding for research has plagued us as a researcher in this space for decades. And again, this comes from the fact that women were excluded from clinical trials, both pregnancy and otherwise. So no knowledge of drugs, of women's physiology is known. And drugs exist for conditions in pregnancy, and this is what the payers will say, but very few are indicated in pregnancy, and most are used off-label, but they're very inexpensive and not overall as effective as needed. Most just treat the symptoms and not the root causes. But the basic fundamental fact is that the drugs that are out there are being used and they're inexpensive. So why should we create drugs that would be expensive, but perhaps be more targeted? And it would seem that pharma would be enthusiastic about closing this gender gap inequality, but it's exactly the opposite for many reasons I just described. So little pharma investment has been in this area. And it's sad. J&J and Ferring are pretty much it as far as the medium to big size pharmas investing in maternal health and women's health at that. And what are the hurdles? Well, I have to say that the regulatory hurdles are a major factor as well. The 2 phase 3 clinical trials that are needed to show efficacy are very expensive. The 2-year follow-up on infants is extremely expensive as well. And in Europe, it's only 6 to 12 months. High-risk populations of fetus and of maternal health and maternal is a complication that, again, liability is great. And the cost to develop the clinical development, as I told you, is a huge expense, and the time to market. And so what I'm hoping this session will help us discuss is, can compromises be made to galvanize the private sector to re-engage in this important public health risk and crisis in the US? Is there a carrot we can offer with the stick? Perhaps accelerated or expedited regulatory pathways, which have been used for this. Can the follow-up of children be decreased in length to some extent? And I know we've had extensive conversations, and I know there's a conflict between sort of pediatric view and obstetrical views to some extent. And can one pivotal phase 3 trial be required before approval, as was with McKenna? and openness to non-traditional clinical trials with modeling. I think that was a really great discussion in the last session by Skip Nelson. I think this is something that really should be done is around the neonatal composite index and what the proper clinical measure would be. And I'll stop there so we can have a rich conversation. Thanks.
Thank you so much, Kelly. And I'm going to remind so many excellent points I couldn't write fast enough. Remind the audience, go ahead and put Q&As or comments in the chat. I can look at them while we're talking. I wanted to start off, Kelly, following your point about, so being here 25 years also, I'm with you that industry and academia need to partner more. And I guess for me though, how do we, for the academics are on the call, we'll leave the FDA off the question for one moment. How do the academics in the room, understanding the return on investment for all of this is going to be delayed for industry, right? Because we don't have biology. Are there easier ways to partner that we're missing? Are we missing opportunities both in pregnancy and women's health to put that carrot out there to industry saying, yes, you're not going to have a drug that's going to be marketable in a year because we need to invest in the biology. How do we pull them in and how do we partner?
Yeah, no, Ferring takes very seriously our external partnerships, and many of those are academics. So I think this is sort of a new model for research and development, at least at Ferring, and I think at many other companies as well. You know, giving that accelerated push to some of the great ideas that are out there. Now, granted, it will come with some ties, but not that necessarily as much as would be. I think we realize the lack of data, the lack of fundamental knowledge, and it needs to be accelerated in this program. I mean, in some sense, this is a very attractive field because nobody else is going into it, but we need to have guarantees and a little bit more involvement with your development. We have many models we're working on now. We have external collaboration project teams that are incorporated into Ferring's own R&D projects. And, you know, I think we don't have a call for information for these grants because they're not really grants. They're more of partnerships. So certainly, you know, reach out to me, reach out to others at Ferring. I think we'd be happy to entertain some ideas, willing to work with you.
That's great. I want to take your second point, but open up to some of the FDAs. So people from the FDA, sorry, not FDAs. So Kelly talked about the 2-year follow-up and other people have talked about infants. And then yesterday, and I think part this morning, losing track of time, we talked about patient engagement. And I know from my patients, from running a prematurity program for 25 years, they would have absolutely accepted a little more long-term risk, potentially, to their babies, knowing they wouldn't have had a 25-weeker. So I don't know if anyone from the FDA can a little speak to this requirement and why we're so off from the EU. And how do we begin to push that boundary? Because I do think, I agree with Dr. Muller, I think it's limiting our ability to do some research for what I think a lot of people would consider safe or acceptable risk. Donnie or Kimberly, you guys one of you guys are gonna have to say something.
I don't know if I'm qualified to say that.
Kevin.
Yeah, I was thinking Kevin would be the one closest.
Okay. Oh, Kevin, then you're up.
I'll try my best to answer the question. You know, it's basically a question that would be better answered by somebody from the review division. But I think what you'll find from the review divisions is that they do exercise some flexibility in some of this, as is with other requirements. The 2 years is traditional, but with a product that has perhaps been clean with no signals in the preclinical space, a lower period of time might be possible. But again, this would be a discussion with the review division. And I would encourage investigators and sponsors to talk with the review divisions about potentially aligning with what the Europeans are doing and hearing what they have to say.
I appreciate that. And Christina, you're going to say something, please.
Yeah, hi. Just to address this point, I just want to highlight that the McKenna trial for the neonatal follow-up really was only 28 days for determining the efficacy, but the 2-year follow-up was conducted under a post-marketing requirement. So, you know, there's certainly flexibility in terms of how much data from the pediatric follow-up we would need pre-market. And I just want to make sure that that point get across.
It does. I think for any of us who have done any follow-up studies, anything beyond even the 28-day window can be quite difficult and is extremely expensive.
Yeah, we certainly recognize that.
Yeah, I appreciate that. So based on a little bit of the thought yesterday, right, about we've done for so long protecting from research, and we're all, I think, a very collective us want to protect through research. And in saying that, based on some of the questions in the chat and some comments earlier, I think this is going to go to Kimberly, but if I'm wrong again, let me know. When we talk about reprotox studies, it strikes me that we're doing stuff that is pretty based in prior ways we did things decades ago, right? We're looking at some very kind of fundamental, is there embryo, is there organogenesis issues? But I think when we talk about pregnancy, there's something much broader, right? We've talked about for the last 2 days how biology shifts. And I just wonder if there's a need to update our approach to reprotoxicology. There's a lot of ways to image how things cross the placenta. And it doesn't seem like any of that is required. So I just wonder your thoughts about leveraging new techniques, maybe changing what we term as so embryogenesis, sure. But when we talk about drugs being able to pass through placenta, either through diffusion or accelerated or facilitated, anyway, so go ahead. Any thoughts on that?
Sure. I appreciate your question. And the studies that I was talking about are pretty much the standard studies to investigate any type of drug toxicity across any space. So those identify risk. However, we do need studies that will look about the potential for fetal exposure. Are these drugs getting across the placenta? And this is very different between biologic products and small molecule products. Both of them reach the fetus in different ways and through different pathways. So I think those studies in cooperation with the traditional drug toxicity studies are very important because we could find that the small molecule or the biologic doesn't necessarily reach the fetus. So maybe there's not as much concern for the fetus in that space, rather than thinking it's an all or nothing type of process. But different, imaging techniques, different ways to look at these things in an animal would really help to translate that process into human subjects.
I appreciate that. I think obviously as we've seen the rise with the novel COVID vaccine, LMPs and other small molecules, the idea of how we study these and how we best serve women is definitely worth a discussion from the reprotox level. In adding to that, Dr. Waldorf asked a question. When preterm birth studies are conducted to test efficacy in non-human primates, what is the expectation of the FDA to show toxicity data in non-human primates? Understanding this animal model is very expensive and access is limited.
Sure. Well, I first want to say that we don't really encourage the use of non-human primates to do the reproductive toxicology studies, mainly because of the expense, the availability, and the fact that gestation is very long and you only have one offspring to evaluate. So you can't really get a good evaluation of risk as you can with a species that has multiple offspring, like the rat or the rabbit. Having said that, there are some products being developed that are only active or pharmacologically active in the non-human primate. So you can conduct what we call proof of concept studies in order to determine in an NHP, is the drug active and doing the molecular mechanism type things that would reduce preterm birth for the hypothesis that you're testing. So we don't necessarily need efficacy in the non-clinical space. We need kind of a proof that your drug is acting in the way that you're intending it to, and then moving forward into the human space.
So to that point, and I'm sorry to keep kind of going on you.
That's fine.
One of the things that I think respectfully I'm not going to go into the progesterone global studies and to so many other drugs that we use in pregnancies, we know the placenta is this enzymatic rich organ. And none of us really know any of the metabolites or the down products, if you will, the downstream products of any of these drugs in the fetus. So how do we attend to that? So the only thing it sounds like the reprotox really looks at is there harm? But There are
Is
so
there
many more
harm?
questions that could be asked. So any thoughts on that?
Right, and I think that is something that investigators kind of need to take into account, that we don't know what each of these products, whether they're approved products or novel products, we don't know how they get to the fetus, and how the placenta plays a role, and what metabolism is occurring. So any investigation of that type of thing is really encouraged to kind of give us as much information as possible in order for us to determine risk.
And I think a bit broader question, not as much about the reprotox, but to the group. So there's I think at least 2 questions in here about pharmacogenetics. So I wonder, just for other members of the panel, your thoughts about how that should get incorporated into these different trials. Donna, you unmuted.
Yeah, I saw that comment in the chat, and I think for pharmacogenomics, it'll be important for drugs that are polymorphic, for polymorphic enzymes that's metabolized by polymorphic enzymes. And in that case, the poor metabolizer versus rapid metabolizer may have different pharmacokinetic profiles. So that will be important, but it'll be important only for drugs that are impacted by those polymorphic enzymes.
So I appreciate that. I put up a slide on a lot of my talks as we don't know what we don't know. And so I'm going to just push back a little on that, is we don't know what has these polymorphic changes because those drugs haven't been studied in that kind of context, at least for the ones that we're using in pregnancy, right? There's some progesterone receptor polymorphisms that Tracy Munnick and other teams stood up looking at this. And those have really, from my understanding, not been looked at as much in how they respond to the trial or not. So just to put that out there. Steve, you had your hand up, or you took it down, or question? You're muted.
How about now?
All good.
He kind of covered what I was going to ask, but I'll ask it a different way. How does the FDA measure exposure in these animals? Do they use dose alone, or do they actually measure blood levels? Because again, these polymorphisms have a huge impact on drug metabolism. So if exposure is measured by dose, that's not a very good indicator of exposure as opposed to a blood level.
Go ahead,
I believe if you're asking for the animal studies, I believe the animal study look at both dose and drug concentrations in many cases.
Thank you.
Yes, they would.
I think maybe back to Kelly, what would need to be shown in order to enter to a phase 1 or 2 trial from the industry standpoint? What needs to kind of be proof of concept from, I think we all kind of have an idea of what academia should be or not, though maybe that's up for discussion. What about from the industry side to buy into seeing if this would work for a therapeutic?
I mean, most assets or companies that we look at, or things that we're developing, we have a fairly good mechanism of action that we know, at least we can show that it has some effect. And I think your comments yesterday too about preterm birth being just, so many different diseases within it and causes and etiologies, that even you identify a specific, whether it be gestational age group or, is it a prevention or is it therapeutic? I think those are different. I mean, we haven't really talked about that. We've just been talking about preterm birth, but many of these things have been used to stop labor as opposed to just prevent. And I think, we'd be interested in something that was prevention based, that had a fairly clear mechanism of action. I think, the microbiome is probably one of the most clear in some ways as far as cervical remodeling. And I think that's an interesting, but then we'd ask the question for the FDA, I mean, are they open to allowing a live bacterial product, if that was the way to go, to be in the vagina of a pregnant woman? Would we have to show bacterial vaginosis? Because bacterial vaginosis may not be what's causing preterm birth. It's the changes in the cervix that are. And so why do we have to go through a phase 3 clinical trial showing, prevention of bacterial vaginosis when we could just look at cervical indicators or preterm birth? And I turn that back to you, Michal. What do you think? And actually, I want an answer from the FDA though.
I'm going to go with the FDA because I think some of the burden for some of the lactobacillus and other, both in pregnancy and even non-pregnant, because I'm involved in that as well, the burden to show that makes no sense to me when that wasn't the burden at all for my fecal microbial transplant. So I'm going to turn it over to, I don't know who wants to take this. Kevin, Kimberly, Christina, who wants to take it? None of you?
I'll take him home. Thank you so much. Because I've heard, I appreciate all the really great questions towards FDA. I think the bottom line is obviously we are not able to provide a one-size-fits-all answer to any of these great questions. As you know, a lot of what we need to ensure that a protocol is adequately designed, both for efficacy and safety, depends on so many factors, right? How well do we understand the disease pathophysiology, understand the mechanism of action? Certainly it goes a long way. You know, what are the toxic findings? What are the, you know, preliminary efficacy findings? And so all of that, and then of course, you know, how does a drug work? And that can drive the endpoint. So I understand, like, everyone wants like a template that we can work from. And we're not, I mean, if this was the 20th program for preterm birth, we can offer something like that, but we are at a very sort of nascent stage. So a lot of this will depend on so many factors driving that program. I do want to mention, if the audience is not already aware, we do have 2 great guidances out there on substantial evidence of effectiveness. And I find those guidances to be very applicable in this program. And it talks about, you know, the traditional requirement for 2 adequate and well-controlled trials, and how various regulations that's, you know, passed along the way, how we have interpreted that. And as you know, we can get to substantial evidence of effectiveness with 2 adequate and well-controlled trials the traditional way. We can get it with 1 very large multi-center trial that basically acts as 2 trials. That's another mechanism. And the last one is 1 single and adequate and well-controlled trial plus adequate confirmatory evidence. So that one has generated a lot of interest in the space of therapeutic development, where it is hard to develop, right? In rare diseases and hard to recruit patients, which I think fits probably the bill for obstetrical indications. So there are definitely regulatory tools that is not just, you know, you have to have 2 adequate and well-controlled trials. So I just want to like put that out there, because I find those 2 guidances to be extremely helpful. So please let me know if anyone needs you know, a link to those guidances.
Thank you so much, Christine. And I want to thank all the panelists for coming on, for contributing, and for helping to move this field forward in all the ways possible. I'm going to turn it over to Nancy. Yes.
Yeah, that's right. Great. Thank you so much, Michal, and for that wonderful panel. I think that it just tremendous discussion we've actually had over the 2 days. So we're going to move into our final session today, which we're going to bring together representatives from each of the sessions to share their key takeaways from their respective sessions, and also for the entire workshop. So I'm going to go ahead and ask our panelists for this to join me on camera. So we have Christy Chang from session 1, Christine Nguyen from session 2. I'm trying to see if everybody's coming on camera. Janine Cross from session 3, Lynn Yao from session 4, and Michal, sorry, we're bringing you back for this one, just got off. So welcome back, everyone. What I thought I would do is start off with, you know, we've heard a lot of information over the last, it really was just 2 half days, even though it feels like it's been 2 full days, I think, of information. It's really been wonderful information shared. If I could kind of do round robin with you, for you to sort of highlight what you felt were a couple of the key takeaways from your session, or the session that you got assigned to, and overall, and I want to just kind of go maybe in order of the sessions that we had. So I wanted to start with Christy, if that's okay. Christy?
Yes, thank you. Yeah, so Kavita isn't able to join us today, so I'm doing this in place of her. So session 1, we had 3 very insightful presentations from Joyce Martin from the CDC, Dr. Selly from March of Dimes, and of course, Michal here. So I'll just focus my takeaway message on the first two presentations, since Michal is here, but she can correct me if I didn't represent her viewpoints correctly. So from the CDC, Joyce Martin's presentation, we learned some very insightful statistics on the preterm birth data on a national scale. We know that preterm birth rates vary widely by states, and they're influenced by demographic factors and access to care. Nationally, earlier in the century, the preterm birth rates were decreasing. However, data from 2014 on showed that this trend had been reversed, with preterm birth rates increasing for all maternal age groups and race and Hispanic origin groups, especially for these early preterm births between 29 to 33 weeks. There are also key subgroups that are associated with higher risk for preterm births, such as multiple births, maternal age extremes, the oldest and the youngest, non-Hispanic Black race, Medicaid or charity payment for insurance, and infertility therapy, mothers who smoke, mothers who have chronic diseases like diabetes or hypertension, as well as previous preterm births. So unfortunately, the CDC data, we're not able to rely on the birth certificates to adequately differentiate between spontaneous and non-spontaneous deliveries. So that's a real limitation for investigators who are trying to conduct research to prevent spontaneous preterm birth. From the March of Dimes, Dr. Salih's presentation, we heard some very sobering statistics that the United States remains among the most dangerous nations where women can give birth. And 36% of all US counties have been designated as maternal care deserts. In fact, more than 2.2 million women of childbearing age live in maternity care deserts. The CDC data are very consistent with the March of Dimes information that 1 out of 10 births in the US are preterm, but there's certainly racial disparity because 1 out of 6 births in African Americans are preterm. And then moving on to Michal's presentation, we learned so many pearls of wisdoms on the biology of preterm birth. Her talk highlighted the fact that after decades of work, there's really no clear understanding of the biological drivers of spontaneous preterm birth. And we really need to explore molecular drivers for both preterm and term parturition. I appreciate that Michal called out the fact that that we haven't been adequately exploring the etiology, the pathophysiology, and the phenotypes of preterm birth, and it's that has limited the progress of research and translation in this field. Certainly, she underscored the need to further understand the biology of spontaneous preterm birth, such as the microbiome of the vaginal cervical space and the biomechanics of the cervix itself. What I took away in an overarching fashion is that the need for safe and effective therapy is really dire, but this really galvanized the galvanizing information really I hope that all the stakeholders will be even more motivated and even more determined to dedicate our resources and our work to the space, because we all are very passionate about improving perinatal care. So thank you.
Great, thanks, Christy. And just as a reminder, I'm going to go around again at the end after you kind of give me your key takeaways, and we're going to talk about ask you for what you've heard for potential next steps to moving towards those solutions, but we'll do that at the end. So let's go around. Christine, I'll go to you next from session 2.
Okay, thank you so much. First of all, I want to thank everyone again for being part of this workshop. It really is so heartening for us to see all interested party, and this is how we start moving the ball. I mean, this happens in many therapeutic areas, so I'm very, very confident it will also happen for obstetrics. So in session 2, we cover the challenges and ethical and regulatory considerations guiding the development of therapies for prevention preterm birth. And I'll kind of go in the order that makes sense to me. We heard from Dr. Liley from UNC on key ethical principles towards a paradigm of inclusion of pregnant individuals in biomedical investigations. And these principles include pregnant individuals being complex, not vulnerable subjects. And, it's interesting, I was looking into what vulnerable means. Vulnerable means that you don't have sort of the independent thinking, the mature thinking to make your own decision, right? Like pediatrics. So that was a very eye-opening for me to actually learn that a few years ago. That change from being complex from being vulnerable to being complex is a huge paradigm shift. Protection through research, again, another huge paradigm shift. And lastly, fair inclusion. And to appreciate these principles, she highlighted the harm of not including pregnant individuals in clinical research. And again, I think this is such an important area to consider when we're looking at benefit risks. The harms of failing to do research in pregnancy includes, lack of evidence on dosing, fetal safety, and maternal outcomes, and the potential risk of undertreatment or toxicity for pregnant people and their fetuses. So we think about, harming pregnant individuals in research. This corollary of harm of not doing research is just as great, if not greater. And excluding pregnant people from research does not eliminate the risk, but rather shifts it to the clinical setting where it may be amplified. Again, another very important potential consequence. I'll save some of the comments about going forward that I heard from Dr. Lyly's research when we come back around. From Dr. Sadeh, he discussed the substantial challenges in with clinical research in pregnancy, as reflected by the clear dearth of research for pregnancy-related conditions. One of the biggest challenges is moving from translational research bench, so to speak, to clinical trial bed. So this is basically moving the knowledge we have from bench to bed. Some other challenges he highlighted were limited funding and limited infrastructure for research. And lastly, limited knowledge to inform selection of potential therapeutic targets and trial design elements. And again, I'll save his comments about potential solutions moving forward in a little bit. And lastly, Dr. Chen discussed FDA's role and requirements for drug development and approval, and in particular for obstetrical indications and for the prevention of preterm birth. She covered 3 categories of approved prescription drug use in pregnancy: on-label, off-label, and those specifically approved for an obstetrical indications. And not surprisingly, the large majority of the use in pregnancy is off-label. She also discussed challenges and opportunities for conducting clinical studies in pregnant individuals, and these include considering the physiological changes in pregnancy and how this impacts safe and effective dosing. This is a very critical consideration in the clinical drug development program. The ethical and practical considerations for enrolling pregnant people in clinical trials, such as informed consent, risk-benefit assessment, and trial design elements. And again, I will save potential paths forward when we come back around. Thank you so much.
Great. Thanks, Christine. That was a terrific summary. Janine, for session 3, you actually, your session had, I think, one of the most impactful series of messages in it. And it was so important, I think, to make sure that everyone is keeping the patient and family voice front and center. So could you share some of your key takeaways from session 3?
Thank you so much. Yes, there were some very interesting, in-depth, heart-filled, and heartbreaking stories that were shared by our panelists. In my session, the panelists include Deb DeSenza, Jen Degley, Ashley Randolph-Cooley, as well as Wakeku Euclid, and myself. Deb DeSenza really talked about the challenges that she had with her infant post-discharge from the NICU. And she noticed that her infant, from her earlier years to toddler years, had developmental challenges that were compelling and posed a lot of struggles for the family. She also had a lot of challenges with the systems and working with providers. to identify some of the developmental issues that were going on, as well as the illnesses and challenges from a disease standpoint for her infant, again, through the toddler years. She continues to see the impact from the NICU course, even into young adulthood for her child. Similarly, Jen Degley also noticed that there was a tremendous impact to her family. And as a parent, she tried oftentimes to protect her preterm infant from exposures to illness. And that's found its way even into school age activities, how she allowed her child to interact with other children, the limitations in the type of activities that her child was able to engage in because of reduced lung capacity and other challenges, but really that need to protect from germs and viruses. Her child got viruses and throughout the childhood course, and it continued to be challenged by these illnesses that her child experienced. One of the key takeaway points that she wanted everyone to know is that the journey does not end at discharge, and this is something that they continue to struggle with. She would like parents to have more information and have providers work on sharing more information, not just to prepare parents for what goes on in the NICU, but also what happens post-discharge as well. She also wants to advocate for engaging parents at the creation of forms, information, and documents, and help to improve the engagement of parents as it comes to information sharing, as well as clinical trials. She also really advocates for any support after discharge, and she feels that that will be welcomed by all parents. Ashley Randolph Cooley also gave a heartbreaking tale of her pregnancy experiences that actually spanned multiple states. She talked about having multiple hospitalizations in pregnancies, hospitalizations in her first pregnancy, in Louisiana. In her first pregnancy, she talked about her engagement with the social services systems, particularly Medicaid and food stamps, SNAP. She had issues ascertaining the type of benefits and the support that she needed. She was told that she could not have those benefits to help support her family. She also felt that as an African American, that she was not supported with specifically having a special needs child. And she was also in college too. And so she was really discriminated against, not just because of her race, but also being a college student. It was almost as if she wasn't looked at as a deserving member in need of benefits while she was going through this pregnancy. She went into labor at 34 weeks in California, and also a 34-week labor in Texas as well. During these experiences, she really talked about being denied access to coverage because of the high costs, particularly with Medicaid. She was denied Medicaid assistance because of her needs, and the medical needs were considered to be too high of a cost. She also talked about the 17P shot was not supported by Medicaid, even after her doctor approved the treatment. She also talked about a very compelling and concerning interaction that she had and involvement with Child Protective Services. And she was interviewed, and some of the concerns was because of her preterm labor. She did not speak to a doctor, and she felt like the investigation, there were some concerns in terms of the quality and the in-depthness and the lack of data that was received by medical providers during her investigation, which really was unfair to her and resulted in a lot of stress, as you can imagine. She moved to Vegas and informed the state that that she had a closed CPS case. But again, those things tend to have a lot of stigma attached to it. And so she had to deal with that as well. In her third pregnancy, which she experienced in California, she said that it was more responsive, but she did not feel heard by providers. So she felt that her medical care was more responsive to her needs, but she felt like she could have been heard more in terms of what her needs were by her providers. She had severe symptoms. She also utilized over-the-counter remedies to find, things to help her and support her. She also had complications attending her appointments due to challenges that she was having. And then she was also dropped by her doctor because, again, a lot of stigma, judgment based on her experience as well. She even had, because of her symptoms started progressing and worsening over time, that she had to be admitted into the emergency room due to severe hydration. She learned that even though she had this horrible experience in different states, that she learned how to advocate for herself. She noticed that, oftentimes the systems tend to overlook the challenges that people face, particularly African American mothers. She also felt that she had to find strength within herself in order to navigate the systems and specifically the NICU. And that speaks to her resilience and the resilience of a lot of mothers that they have to really demonstrate in order to get the services and the support that they need. She also talked about Glow Preemies as a source for African American parents to help address their challenges. And this is also a resource that can be very beneficial to African American families. We also talked about in our session community and system level impact of preterm infants. And Wakeko Euclid really talked about the impact on families and that impact being multidimensional, really impacting psychosocial, financial, mental, emotional, physical. And a lot of it was time intensive in terms of experiences for families. Families may not know what to do after their NICU stay, and that can pose additional stress and concerns for them. And there may not be a lot of the support that's needed for preterm infants. There are also an increased cost burdens to family. And again, there's a lifelong impact to families, especially when we're talking about infant mortality, changes in family dynamics, long-term psychosocial impacts on families as well as communities. And the most important resources is for family-led NICU support during and post-NICU stays. Because they have experienced the help and support sometimes in the NICU, but not so much post-discharge. And then my summary, I really talked about how very challenging the NICU system is and really learned, hopefully we understand and appreciate the complexity of the systems with the interdisciplinary providers. All of them with the same goal, but not always with the same pathways to that goal. This is my phone ringing. I'm so sorry. My phone does not ring no time, and then I'm doing this, so I apologize. Same goal to get infants discharged after treatment. Infants are not the only patient in the NICU, was my point. We really have to look at the family system as their parents, as the family system as the patient as well. And we want to make sure that we partner with our parents and families and bring them onto the NICU team as much as possible. Policies and procedures that are developed to govern and regulate the system, they're also sometimes stressor for families because they're not always in the best interest of the families. In terms of governance of the flow, that's what they're there for. They're supposed to help with the workflow, but sometimes they add stressors in terms of limitations to visitations, interactions, and so forth. And sometimes they make sure that these policies are centered around the staff rather than the patient needs. And sometimes we don't center the patient and the family when we consider these policies. Additional systems, families also have the burden of having to deal with not just the NICU system and the healthcare system, but oftentimes they're dealing with insurance coverage, Social Security benefits, that adds to complications and stressors for the families. We are really asking them to understand a very complex system in a very short period of time, dealing probably with the most stressful situation of having a sick child. And oftentimes the mothers are recovering from their own illnesses in terms of traumatic deliveries as well. The NICU and the healthcare settings are embedded in the systems in which in the neighborhoods, the context of the neighborhood and the environment. And so a lot of those social issues find their way into the NICU as well. And that includes, poverty, intimate partner violence, social risk other types of social risk factors such as homelessness and substance use as well. And so we need to be aware of these social issues. And so these families have to not only deal with having a NICU infant and all the medical concerns, but they're also the social issues of poverty and violence as well. And so social determinants plays a role also in the NICU course. There are NICU programs to help support families. With all that being said, there are many support systems in the NICU and outside of the NICU, community supports that we use to support our families. They are not all the same. They are not all equitable. Some NICUs have psychologists, for example. Some NICUs do not. Some NICUs do not have social workers, and some do. And so we even though we do have supports in the NICU, they're not all the same. And so oftentimes we do have to rely on community services for substance abuse support, mental health support, housing, violence assistance and support. And these partnerships with CPS are oftentimes what is needed to help avoid some of the negative consequences that can occur because of involvement with the CPS system. So really encouraging our NICU to reach out into the community and really develop very strong partnerships with all the systems that we have to engage in to best support our families in the NICU. Thank you.
Okay, great. Thanks, Janine. And then I'm going to turn to the sessions that we had today. So those might be a little fresher in everyone's minds since we just had them. So Lynn, you want to sort of give some highlights, key takeaways that you heard from session 4?
Absolutely. And it's hard to come after Janine, I'll say that. But
yeah.
I will try to just give a pearl or two from our speakers. First, Mark spoke. As a neonatologist, he had a lot of experience about, what the specific endpoints, why McKenna failed. Gestational age alone isn't the best predictor of outcome, but he did point out that every week that you are able, you prolong not delivered, decreases morbidity, decreases mortality. And his pearl, I think, was, to analyze neonatal outcomes potentially by distribution curves of, survival and morbidities by gestational age to come up with potential composite outcomes. So I thought that was a really good point. For Tamara, again, I think her message was loud and clear that we need to keep families and patients front and center in whatever type of endpoint we consider. And that there is this need to think about nuanced endpoints, not just necessarily something that is more easily measured or more necessarily objective if we actually miss the point of what really matters. I think Anjali's take-home points, a couple of them I think were really important. We need to think early about how this treatment would be accessed and used, even if, if it becomes available. Is everybody going to have access or equal access to the treatments when they become available? And we have to think also feasibly about what measurement in terms of length, in terms of follow-up, and in terms of complexity that we can really realistically expect for centers who would be conducting the research, which may very well be community-based NICUs, to successfully conduct the actual studies. Christy and Ann, I'm going to put together, and I think the major points there were that FDA would love to have an easy answer, and we know that it's not so easy. I think Ann came up with some ideas about this global rank scale, and that's already under consideration with the INC, something that can provide at least a little bit more information than a binary yes or no outcome, clinical outcome. And then finally with Skip, I think Skip made a couple of comments that are really important, including that, he's wondering as a global issue, whether or not the science is there enough and actionable enough that we're going to have any movement, because if we don't have actionable science to target, then are we going to get anything studied? And I think that's a really important question to ask. And he also came up with the idea of, if we're going to come up with outcome measures that are nuanced, that maybe are continuous, and that maybe are a little bit more reflective of actual clinically important outcomes to patients and families, that we're going to have to take a look at the data we have, and perhaps what we need to do is model it and collect the data and see what do those outcomes look like. Maybe they're based on gestational age, or maybe they're not, but we've really got to do a better job, I think, of collecting the data and using some, hopefully, modeling and simulation techniques that can get us to the answers. And then I think I'll stop there, because we'll talk about path forward, and I did get some ideas from the speakers as well.
Okay, terrific. Thanks, Lynn. Michal, I'm going to go with you, and actually go ahead and give me your key takeaways and your solutions, if you will, or your key priorities for next steps. And then I'm going to go in reverse order for solutions. So I think maybe that's a fair way to go about it. So go ahead.
All right. Well, again, thank you, everyone. And, this is something that's really near and dear to my heart and has been. So I'm a physician scientist. I ran a prematurity prevention program for 20 years. And I have to tell you, for all the stories we heard yesterday, I heard those over and over again every Wednesday afternoon. And for the women with subsequent pregnancies who would then say to me, so Dr. Elvitz, what are you doing this time? So over the years, I offered cervical length screening. I offered McKenna. The cervical length screening would be like, okay, how good is that? For McKenna, well, why does it work, Dr. Elvitz? I don't know. How are you going to prevent it this time? I don't know. Have your studies shown anything? We're trying, right? So we've all been doing this as a community for decades, and we have to be honest that we haven't made a lot of advancement. And I think we need, and I'm so glad we're having this webinar, to ask why. And I want to be clear that we, as a doctor, as a scientist, as a patient, all want to do more. We have to stop that urge from wanting to do more to have us do things that are not effective and helpful for the patient. And I think that gets in our way a lot. So there's been this focus, to sum up kind of our meeting, there's been this focus on protecting pregnant individuals through research as opposed from research. I think what we learned both in session 1 throughout the day and especially at the end of the session today, there are many obstacles. So I'm going to list 4 of them. The first one, lack of investment has resulted in a lack of discovery and a lack of being able to leverage innovative research in the field of pregnancy health, and specifically in preterm birth. As such, the very biology that drives pregnancy health and disease remains mostly unknown, which means every other discussion that you're hearing is predicated on not having that knowledge, which leads us to the difficulty both between the FDA and trial design, because we're acting out of a vacuum. So that's number 1. Number 2, the current FDA requirements to protect from fetal risk are really important when we talk about impact on embryogenesis. And I do wonder, as a daughter of DES myself, how much DES and thalidomide continue to cloud. I'm not saying they shouldn't influence, but continue to cloud the way that we think about pregnancy health and using therapeutics. And I do wonder if that's worthy of a different discussion that we didn't have today. I think a lesson from session 5, consideration of how small molecules and biologics, which are being used in every other discipline across medicine, influence and should be used, or how they create reprotoxicology or contribute to that. We really need to start thinking about in optimizing maternal health so we don't lose the opportunity for these therapeutics to improve health for both mom and baby. So that was number 2. Number 3, as Dr. Moley and others alluded to, the lack of productive partnerships between industry and academia, with the FDA taking an integral part of that, is really an obstacle to us moving this field forward. And I really do wonder how much it's worth engaging the patient perspective, as we just heard from Janine and others, in what is acceptable risk. And maybe it's time to redefine that for pregnancy and maternal and child health. Number 4, clinical trials done. So let me preface this because I'm about to get on a big soapbox. I so appreciate the NIH, FDA, CDC, March of Dimes, Gates Foundation, everyone for elevating clinical trials. But I do have to say that we've made a humongous misstep. Clinical trials done with therapeutics that do not target biological, plausible mechanisms have cost a lot of money and have not advanced care at all. So we can talk about McKenna, the initial paper on McKenna in that New England Journal of Medicine article. The reason it was used, it says, because we believe it affects myometrial contractility. The clinical trials that are ongoing right now with PESRI are because we think they prevent cervical remodeling. None of this is based on biology. And so we're putting a patient on burdens and on cost and we're not advancing health. So in saying those 4 obstacles, next steps to me are we have resources. We have little ones, not as much as some other fields, but we do have resources, but we need to invest them in a different space. I would offer that we need to stop doing clinical trials on any therapeutic that does not have a consensus that it targets a biological mechanism involved in the pathogenesis of preterm birth. And all those resources currently happening in clinical trials should go to biology from all the different funding sources, number 1. Number 2, we should mandate how we define preterm birth, not just spontaneous and medical, which has obviously been a miss, but we need to capture metadata so we're able to bucket the different kinds of spontaneous preterm birth clinically, so we can then equate that to what we're seeing biologically. We need to mandate, along with preterm birth phenotyping, no clinical trials without biological plausibility, partnering of scientists, clinicians, and patients in how we approach research, how we design trials, and what we deem acceptable risk. Steps off soapbox.
Great. Thank you so much. So I said I'm going to go in reverse order. So I'm going to come back to you, Lynn. And I realize as we go through these, we're probably going to have some repeats. So if you want to expand on something that the prior person said, that's fine, but or add to whatever you have on your list. And this would be sort of key things you heard from your sessions as high priority next steps.
Yep. Thank you, Nancy. So again, high priority next steps from from what I heard from our session. I will say that, I take what Michal said. Absolutely. I think well said. I think there was a sentiment in the group that, we need to really think hard about where we put our dollars and where we put our research, but we can't stop everything until we figure it out everything, or it's going to take too long. So I did hear a call for, continuing to figure out what we have that's best at the time while we're gaining extra data that will help us. So I think that was one important thing. I think that in the question that we were being asked, which was, how do we look at clinical outcomes? What could we consider? I think what we heard is a strong call to try and look at a different way to think about outcomes in neonatology or in the neonatal outcomes, including thinking about sort of not necessarily binary composites, but continuous variables or ordinal scales, and that research into that and coming up with those types of endpoints would be of great value. I think what we heard was also that we have to incorporate the patient voice and the family in, not necessarily that it would only be a patient voice endpoint, a patient reported outcome, but that the weight of whatever composite is being incorporated into this, into an endpoint, has to be weighted based on what we hear from families and patients, and that type of weighting is critically important. And then I think finally, gestational age, although it's very easy, we were bashing gestational age a bit, I think, during this meeting. But gestational age it certainly is not the only thing, but it's still absolutely important. We can't forget its importance in understanding prematurity and the consequences of prematurity. So that's sort of my summary of next steps.
Okay, terrific. Thanks, Lynn. I'm going to come to Janine.
Yes, thank you. For my session, I think some of the next steps that I think we really need to really think critically about is, thinking about how we support our families and what it looks like. From our session, a lot of the parents talked about, being surprised at the post-discharge complications and the problem. And so I think some of the innovation here is when we're doing patient education and teaching, one, it's something that should be happening from the very beginning up until discharge, not just waiting until discharge to do this, but really thinking about what other providers and specialists to bring into the NICU as part of that. For one, it could be developmental follow-up specialists to help have that conversation with families about what to expect. Have pediatricians in the community who takes care of preterm babies and talk about some of the challenges that families can face with illnesses and challenges that way. Also really trying to streamline policies about proper mental health support, particularly to address anxiety, perinatal mood disorders such as anxiety, depression, and trauma. Really being innovative and maybe thinking about maybe substance abuse counselors or substance abuse specialists or peer support people in the NICU to help support families as well. Also, you know, from what one of the participants talked about, her experiences with racism, discrimination, biases, really thinking about, you know, what type of DEI, diversity, equity, and inclusion programs need to be incorporated in the NICU, but not just that, but now how do we educate the community and the systems about what it looks like to be a family in the NICU and to have a NICU infant, you know, really educating insurance systems, bringing all the stakeholders to the table and make sure we really understand what a NICU experience looks like and what the impacts are on families, including child welfare services, CPS, so that they really understand and they can ask questions from the experts so they know what they're looking at. I do know that there are some models that have really moved the needle on this, where I know CPS have now employed nurses so that when they get reports and investigations, they have some kind of clinical medical professional that can read that report and say on an investigation and say, hey, well, you know, with these eyes from a clinical standpoint, you know, this is expected, you know, these concerns are expected. And so again, maybe making it more streamlined to have healthcare professionals with NICU experience with infants and families to partner and work and be consultants to child welfare services to help support the needs of families. And also really thinking about, you know, global, you know, state policies, and really understand that some of the concerns that this one particular mother had about, you know, racism and discrimination, health disparities as well, it really makes it clear that it's not a one state issue. This is national. We have a problem. We have a national problem with this. And so if she had this and it's an isolated incident, great. That's not what was conveyed. Jump states, same issues, same challenges, not being heard. And so we really, it's a call to action and awareness to NICU, health settings, and the systems in which our families have to work from. We need to do better. We need to do better. And also, we have guidelines, we have recommendations, we have guidelines, we have policies, they're out there. A lot of the organizations that I talked about in our sessions, the National Perinatal Association, National Association of Perinatal Social Workers, March of Dimes. We have organizations that have put in the work to say, hey, we know what to do. And I want to strongly encourage, you know, NICU healthcare settings to really, you know, find the resources, do a needs assessment, not only with your patients, with your staff, but do the work as an organization and find the areas in which you need to improve. I think some of the best healthcare systems have patient advisory boards. They really reach out to patients and say, hey, what could have been, how could we have served you better? And incorporate their feedback and ways to support them the best way. So again, working together with families, helping families see the progress and supporting them in their progress, having clear communication to ensure that families understand what is going on, not just during the NICU course, but after, celebrating all the milestones that the family experience with the child. And families need to feel and feel supported, again, not just during the NICU course, but after. And then we really need to understand what support looks like and make sure that support is equitable to all, regardless of background, ethnicity, spirituality, and so forth.
Great, terrific. Thanks, Janine. Christine, coming to you.
Great, thank you so much. I will try to keep it brief. So from Dr. Liley's presentation, I would say what I call my key words is that research in pregnancy is an ethical imperative. I think that we just have to say that over and over and over again, because that is a huge paradigm shift. It really is when you think about it, right? Because usually within ethics, pregnancy research, ethics there is to put up roadblocks. They do not. And in fact, they're there to guide the safe and effective evaluation of therapies in pregnant individuals. They are really an effective tool when we apply it the correct way. So first and foremost, though, I think we have to make that principle loud and clear. It is an ethical imperative that we get the data we need to treat our patients right, because they're pregnant. In no way put them as second classes, and by not getting the evidence they need, we essentially are doing that. So I think we have to keep that front and center. And it's out of the basic rights of, you know, individuals, pregnant or not, deserve protection, access, and respect. And I know we sang that tune many years ago for pediatrics, and it's about time we make sure that applies to pregnant individuals. From Dr. Sede's presentation, one thing that really caught my attention was for his potential solutions, he mentioned a lot of different regulatory tools to help move, you know, either to help fund or support, expedite research and clinical drug development in the space. So I would like to reassess what FDA is doing on our part to educate the community, the research community, and even, you know, our regulated industry on how these tools could be utilized and relied upon. I think there's more work to be done on our part for that. Regulations are very, very hard to understand. And so I think to the extent that we can get out there publicly and actually educate the people who may be impacted in, you know, research and pregnancy or funding, to understand how they can best use these tools. And that's what they're for. They are really there to help programs that are hard to develop. And lastly, Dr. Chan discussed the regulatory considerations. I want to go back to the benefit risk framework that we consistently use when we evaluate our market applications review. We talked a lot about patient voice. And it's really important to understand that we absolutely value the patient voice. We put that as part of our framework, but our framework also has to consider the benefit and risk, right? By the time we get a drug to the patient that is FDA approved, it has been vetted, it has been evaluated. And that's, you know, that is our mission, really, is to, when we approve a drug, the patient, their family can be assured that the drug works and it is safe. The benefits are such that it can offset the risk, because, as you know, all drugs have risk. I mean, there's no drug that's risk-free. So that framework is very important in how we make our decisions. So it's not like we're ignoring patients' voice at all, but there's this framework, and there are other key factors that we need to consider before we can approve a drug. So thank you.
Great. Thank you, Christine. And Christy, I'm going to come to you. I know we're running a little over time, but you're going to have the last word for today, for the session.
Well, thank you so much, Nancy. I just wanted to thank everyone for attending. So I'll make my remarks very brief. 3 takeaways, moving forward, 3 takeaways. So the first one is I'm hoping that this workshop is a great start to get the conversation going between all the stakeholders. And secondly, I'm encouraging our researchers, industry, investigators to pick the low-hanging fruits while we're working on the biology and mechanism of preterm birth, spontaneous preterm birth. Please gather some PK information from women who are already taking various products, you know, when they're pregnant. There's no safety concern or no ethical concerns because these women are already taking these products. So please, you know, gather the data from there. And then lastly, as a review division, we are open and available and willing to meet with any stakeholders for pre-IND advice and consultation. So I would encourage everyone to take you know, to take up the opportunity we have to meet with them. So thank you.
Terrific. Thank you so much. That was a great way to sum that up, Christy. Thank you. So thanks, everyone. With that, we have reached the end of our outstanding 2-day workshop. And I think, as everyone is well aware, and we've heard even many of the sessions, including just what Christy mentioned, this is not the end of the discussion on this important topic, but rather an important step in moving the field forward so that new therapies can be developed and ultimately marketed to prevent spontaneous preterm birth. Duke-Margolis and our colleagues at the FDA are committed to working on this critical issue. Before I go, I want to take a moment to thank everyone who contributed to this excellent workshop. First, thank you to all of the speakers and panelists who shared their expertise with us over the course of the workshop, and especially the parents who shared their personal experiences. I'd also like to extend a deep gratitude to our colleagues at the FDA's planning team for this workshop, including Christy Chang, Christine Nguyen, Janet Maynard, Catherine Pilgrim-Grayson, Leela Sahin, and Lynn Yao. And a big thank you to our Duke-Margolis team, including Duray Kim, Beth Boyer, Cameron Joyce, Mia Williams, Hannah Vitello, and Luke DeRocher. Finally, I want to thank our audience. You have contributed so many wonderful questions and comments and really engaged in this conversation. And it was really wonderful to have your attention and engagement. So we really thank you for that. And with that, I wish you all a tremendous, wonderful rest of the afternoon. Thank you.